Esto lo abriste directo desde la aplicación, digamos. Tendría que haber estado ahí la sesión ya creada.
Claro, entré la aplicación.
Chau.
Sí, ese está aquí.
La mierda, claro.
Deberían de haber hecho otro Zoom para hacer pruebas, ¿sabes? Con la chica.
Claro.
Y luego este es el del bolo. ¿Sabes? Quiero ver una cosa, vale. La Merced, todo el retorno del Zoom. Creo que me cabe.
Sí,
que sale la tarjeta.
¿Cómo se nota que esto lo conectó Mercedes?
Sí. El 9. Vale.
Pobre Mercedes, o sea...
Me tuve que llamar, ¿eh?
Me dijo si me podía llamar, si pasaba. Sí, no sé. Y al final te
cambio aquí, es este. Hola. ¿Qué tal? Buena tarde. ¿Tot bé?
Sí. Ara la ministro,
¿No era la 1? Ah, vale, es que había iniciado el Zoom ya. Vale.
Bueno, no, déjalo, sí.
Vale.
Pero es a la 1:30, a la 1:30, que dijo la... Como ayer lo pudimos, porque...
Sí.
What move our field forward. Let's begin. Now I pass next speech will be done by Carlota Villar, Vice President of Barcelona PID Foundation. Thank you, all of you.
Thank you, Ted.
Thank you very much. Good afternoon, everyone. I'm Carlota Villar. I'm the Vice President of the Barcelona PID Foundation, and I'm mother of a boy with a PID. It is a pleasure to welcome you today to the meeting Molecular Insights of Diagnostic Challenges in CVID, held within the framework of our PID education program, which aims to promote education, knowledge sharing, and continuous improvement in the field of primary immunodeficiencies. As a foundation, we strongly advocate for excellence in research on primary immunodeficiencies as a key driver for progress in the understanding, diagnosis, and management of these rare diseases. We would like to express our gratitude to our sponsors, Seguida and Advanced Diagnostics, for their support and commitment to education and research, and to Vall d'Hebron University Hospital and the Vall d'Hebron Institute of Research for hosting this meeting. Thank you very much. We are also grateful to all the speakers for sharing their expertise, and to all participants joining us on site and online for being part of this session. We hope you will enjoy the excellent presentations prepared for this afternoon, and I'm sure that they will lead to a very interesting discussion. Thank you very much and welcome.
Thank you, Carlota. So, yeah, this event will be divided in different sections. First, we have a bit of an educational overview. Then we'll have a section to discuss unexpected models that can underlie the genetics of common variable immunodeficiency. And then we'll go a bit farther and explore these challenging mechanisms that can be underlying the disorder. And finally, we'll have a roundtable. And we expect all of you to participate, both here in person and also online. You can use the Q&A section of the Zoom meeting, and we'll be controlling the questions so you can all participate. By the way, I'm Laura Batlle Masó. I work here at Vall d'Hebron Research Institute at the Immunogenetics Lab, and I'm also in charge of the educational section of the Barcelona PID Foundation. And with that, I will pass the word to my colleague.
Thank you, Laura. My name is Jacques Riviere. I'm a pediatric immunologist in Vall-d'Hebron Hospital. It's my pleasure to be here and to co-host the meeting with Laura. So today we're going to have, as Laura said, great speakers, and we can start with the first of them. But thanks, I want to thank you, both the Barcelona PID Foundation and the VHIR institution for the welcoming for all the participants. So it's really my pleasure to introduce Dr. Pang. Dr. Pang is an assistant professor of pediatrics and genetics in Montefiore Einstein in New York. She also served as director of Advanced Diagnostics at the New York Center for Rare Diseases, and she's an excellent scientist, a beautiful mind, really, and a really good person. So I don't know if the connection is already. She wasn't able to be in person. But she's connected, and she woke up early this morning to be able to do the talk. So thank you, Xiao.
Hey guys, can you hear me? Yeah?
Yeah.
Okay, great, awesome. So I'm gonna get started because I have a lot of slides, and I apologize beforehand if I talk very fast because I'm a New Yorker. So thanks for having me. It's great to see everyone virtually. Wish I could be there with you in Barcelona instead of in this lovely -8-degree weather in Pittsburgh. And I'll try to join the roundtable after if possible. I'm actually in transit from another conference, so bear with me. Okay, so there's going to be a lot of talk about CVID today, and I think one of the first things that people always ask me is, is CVID a genetic disease? And, I'm a geneticist, right? And so one of the things I always try to say to people first off is there is actually no real thing as a term, as a genetic disease, because all of our human diseases have some kind of genetic contribution, right? Just like all of our diseases have some kind of environmental contribution, and some roll of the dice from chance. So I think the reason that some of us are so drawn to the study of human immune disease, though, is because, of course, the way in which immunity processes interact with the environment makes it so that it often plays an outsized role in determining how and when those genetics manifest. And I think you'll hear from all of the subsequent speakers some really, really great stories about different kinds of mechanisms that sort of explain that. I'm really here to sort of give you guys the broad overview of I guess you could say, the evolution of the understanding of what CVID means as a label, because so much has changed, I think, in the last few decades of medicine. So I hope if you have to walk away with one message, it's that if anything, CVID is not really even a one thing, right? It's a very, it's a lot of different kinds of diverse genetic architectures and landscapes. There are genes associated with CVID that actually span the entire realm of effect sizes and of allele frequencies. And certainly it's not one pathobiology with every single individual that sits in front of you in the clinic who has that particular label. So you have to really think carefully for every single individual patient. You have to think about what their actual underlying drivers of disease are and treat those. A lot of how we began to think about CVID is also coming from a place of many assumptions and many forms of ascertainment bias, okay? And it's always really important to stay humble. Our patients are always going to teach us more than we have to teach them, because think about how big human biology space is and even the size of human genetic space, and think about how little we actually have in the way of medical assays and in the way of things we actually know about disease, okay? So never say never. One thing I really want to emphasize, and I'm trained historically as a yeast protein biochemist who's never taken a single human genetics class in my life. So what right do I have to be sitting here telling you guys about the genetics of human immunity? Well, I think one thing that kind of always humbles me is the evolutionary perspective. And from the evolutionary perspective, all living things have some form of immunity, because all living things have to fight this battle of host versus pathogen, right? And certainly in humans, we have learned through the discovery of many, many, many genetically driven forms of immune disease by now that if you have impaired host factors, this leads to increased susceptibility to infection or foreign growths, right? And if you have dysregulated host factors, this leads to, all sorts of uncontrolled, spontaneous, overexuberant kinds of parties in your body, people getting fires set by things that shouldn't be setting fires. So one thing that's important to always think about when you see a new patient is this, which is that immunity is not a bunch of cells. It's not even an organ system. It's a series of processes as old as life itself. And we have immunity in all of our cells. We just ironically had the best assays available to interrogate the most evolutionarily recent and smallest part of immunity, which I, call the professional immune cells for my patients. And those were the oldest. And so those were the things that we were able to know anything about earliest in at least the history of the study of genetically driven immune disease. Now we're starting to have great emerging clinical assays that enable us to understand a little bit better about innate immunity. And we still have very few, if any, clinical assays for cell intrinsic immunity. And so that any understanding of the existence of those diseases has really relied upon advances so far in genetics and genomics. But when we think about CVID, we still need to think about this entire chain, all of these different mechanisms. So in order to sort of step back a little bit, I think we need to understand where we are right now in the history of medicine. Historically, we really just had to do one thing. We had to match some, labs and some symptoms and come up with a clinical diagnosis, and then we could pat ourselves on the back and congratulate ourselves, right? The patient has CVID, put them on immunoglobulin replacement, maybe, some infection prophylaxis, we're done. This is no longer the case, right? Because now we have molecular diagnoses, we have cellular and biochemical phenotypes that we now have to match these clinical labels. So now we not only are having to conceptualize our patients at multiple different levels of thought, we're also having to diagnose them on multiple different levels of organization and etiology and try to reconcile these. So, what geneticists basically did before the advent of, molecular diagnostics is we were art historians. We were sitting there matching ears, matching eyes, matching, hands, and being like, oh yeah, this is more likely to be a Bellini if it looks like this, and it's more likely to be a Mantegna if it looks like this. And, if you're lucky, the diseases you were looking at, there was, some quite consistent, robustly penetrant, and easily recognizable kinds of features, and you could be right. Just based on the number of misattributed paintings, which are static and two-dimensional in art museums, I think you could probably figure out that this is pretty challenging to apply even to painting, much less to human biology, right? So we're very lucky to, have begun to have assays that enable us to sort of look under the hood of the car to understand maybe what's going on underneath. And one of these, of course, is, genomics. So this is a very recent phenomenon, and I hope we all continue to feel lucky about that, right? It was only about 20 years ago that we had any opportunity to even know where genes were, what they looked like, and how they sat in relation to each other in the genome. It was only about 10 years ago that people really began to be able to use this kind of information clinically. Someone hands you a result and says, hey, okay, now go and treat your patient, right? So that means we're still very much on the steep end of the learning curve, okay? And there's still many limitations to genetic testing, and we should remember that it does not contain all of the answers. It is simply yet another assay that we use to help us take better care of patients. How has the advent of widespread molecular genetics been able to help us rethink human disease, though? Well, one of the most obvious things is it's expanded the genetic landscape of what our known clinical phenotypes has been, right? Like CVID. So, this is from the review that Andres and I wrote, and you can see back in 1971, basically a bunch of people got together at the WHO, and they were like, okay, we know all of 5 discrete genetically driven immune diseases. So those have names. all these other ones which have no names and which, based on our available assays, which mostly detect humoral immunodeficiency, we don't really know what they are. We think that they're going to become, some kind of discrete entity down the road, which, there's a lot of foresight to realize that. They're like, let's just stick it all into a bucket called common variable immunodeficiency, right? And that, sadly hasn't evolved that far. I mean, for a good, few years afterwards, people were trying to use all kinds of different clinical features to try to figure out, well, which of these people who got that label would cruise on IGRT and which ones would continue to go on to have all kinds of immune dysregulatory and inflammatory complications. And it wasn't really until the advent of novel methods for interrogating different immunity processes and interrogating different kinds of natural history data that we were able to begin to have any traction on those different kinds of biological endotypes that exist within CVID. And one of the biggest services, of course, came from the fact that people started to identify monogenic etiologies underlying some of these clinical labels, right? And you can see from just these genes here how different some of these processes are that they were finding mutations in. And just if you mapped where these particular genes' protein products are expressed and what they do, you can see that a lot of their functions certainly span the entire IUIS nosology. But the thing that is most striking is that most of these are not specific to B cells. They're not B cell intrinsic limited functions. They're not necessarily specific even to lymphocytes or adaptive immunity. And we'll come back to this as well. Okay. So in addition to, of course, expanding the genetic landscape, we've also expanded the kinds of allelic mechanisms that we know to be associated with some of these known immune diseases and immune disease genes, okay? So here's an example of a patient that I took care of when I was working at Hopkins, which and came to me already with like 4 different clinical labels, and they didn't consult us until he had already exploded his gut twice, okay? So there's another thing, consult your geneticist earlier. But he ended up, I was handed a result for him. And the result simply said, okay, that looks like there's a likely pathogenic de novo, presumed de novo IKZF variant, okay? And everyone was like, oh yeah, he has CVID. Because at the time, this particular first form of IKZF1 mutation mechanism, that was the only one characterized in OMIM, okay, cataloged in OMIM. And, OMIM is a very, very well-respected, resource, and so everyone's like, oh yeah, that must be it. Well, if you actually look at the Icarus literature, turns out there's like at least probably 7 different forms of loss of function, gain of function, and dominant negative kinds of allelic mechanisms. And this person actually had one of the ones that was a gain of function form. And because it was a gain of function form, this meant that it was potentially tractable to different kinds of therapies. This is why it mattered, not just to identify the variant, but to understand how it was causing that person's specific disease, even though it sounds like from a first pass kind of comparison that really the CVID form could have fit. But because of this particular gain of function, we were able to try to use thalidomide derivatives to try to degrade the protein and sort of target that particular specific mechanism that way, okay? And as it turns out, of course, subsequently, a lot of people, Sergio and others, went on to show that, it turns out it's not just that one dude who had this diagnosis of, a clinical label called IgG4-related disease, but a lot of people who get this particular form of Icarus mutation also evolve into getting IgG4-related disease, yet another diagnosis of exclusion, right? So now we're really starting to map underlying molecular diagnoses onto different kinds of clinical labels. Certainly, this has also reorganized how we think about, how these particular ones are related. It's reorganized how we think about basic illness scripts of what we call our known monogenic disorders, right? And nowhere is this, probably better as an example than the RAG1 and 2 deficiencies, right? So, Gigi Nolarangelo gave a great talk at ESA 2 years ago about this. When we think about RAG1 and 2 deficiency, we don't immediately jump to thinking, oh, this person must have SCID anymore, right? Because the person can have anything along the spectrum. And it makes it actually harder when they don't have SCID to think, well, when am I going to transplant this person? How am I going to transplant this person? What other things do I have to worry about for this person as potential complications, even going into transplant, right? So these are things that really change how we think about managing and diagnosing patients. More than just classic PIDs, kind of, exploding paradigms, we have so many examples of immune disease where just having less of something isn't necessarily better, and just having less of something doesn't simply mean you just have milder disease or later onset forms of the same disease, which is what a lot of geneticists are often taught about other kinds of conditions, right? But, Andres is going to tell you all about TASI. TASI is all over the place, right? And certainly if you have some degree of residual TASI signaling, doesn't mean you just have certain, obvious forms of immunodeficiency, or even, the same form of broken immune tolerance all the time. If you have no otulin function, you're going to have sterile, early onset, usually, pretty severe systemic autoinflammation. But your parents, they're probably going to not have anything, or they're going to have the occasional Staph aureus infection, which can then lead to infection-induced autoinflammation. And I think everyone knows the example of X-linked CGD carriers, right? Because you can't sit around twiddling your thumbs when you find an X-linked CGD mom. You really have to worry about, are they going to develop type 1 interferonopathy? Are they headed down the path to lupus? Are they going to have, CD21 low B cell expansion? So, I usually watch these patients like a hawk, just waiting for them to sort of develop something. And that has nothing to do with, whether or not they're able to be, defend against infection, because it's a different aspect of, residual NAD/pH oxidase activity. So yeah, management, right? This is why we care. We want to manage our patients better. And certainly when we know more about their genotypes and phenotypes, we can try to do that. So nowhere is this, more true than in ALPS land. I get a lot of autoimmune cytopenia and ALPS consults from our hematology friends. So, the classic one is always, can you help us send an ALPS panel, because I think my patient has ALPS? And my question for them is always, well, are you talking about clinical ALPS? Do they have the clinical manifestations that look like lymphoproliferative disease to you? Or do you really think they actually have primary genotypic ALPS, which is a very limited subset of, etiologies in that realm? And so we have to always think about really carefully, right? Because if you choose to do an ALPS panel and the person turns out to have NF-kappa B1 deficiency, like in this particular case, you kind of, picked the wrong test, right? And, a lot of times the way we know that is we ask questions about what other stuff do they have besides the actual lymphoproliferation, whether there's stuff that their family members have. And, particularly with NF-kappa B1 deficiency, which is a very common monogenic disease that I think we all see, this can present as all kinds of things, right? It can certainly present as CVID, but it can present as all these other things too. And you don't want to miss the one that presents as first break lymphoma. Same thing here. This is actually a giant family of ALPS patients I took care of, none of whom had ALPS, right? And then, what do you do in that situation? Well, you try to treat them with the appropriate medication and not the default knee-jerk empiric one that the hematologists want to give them, right? So one other thing that I don't think we talk about enough, of course, is how much we've learned from our patients who have monogenic disease about how we can use this information to also help us take care of undifferentiated or presumed polygenic architectures of disease. And nowhere is this more true than for diseases like lupus. So I have a buttload of lupus patients, and they all are quite different in their underlying biologies. And one of the ways we know this is because lupus is actually one of the diseases where we started off very early finding monogenic correlates, right, in the early complement pathway genes. And since then, we found all of these different kinds of pathways and processes that can lead to a person who gets a clinical label of lupus. And actually, the biggest growing subset is none of these. It's actually immunometabolic diseases, like people who have mitochondrial disease. So, we're constantly discovering new ways in which a person can end up with, the same kind of surface surface label. So not to belabor the point, but, I think we've done this relatively well for some conditions, okay? And other labels, I think we're just starting to get any degree of underlying molecular traction on, and hopefully over time we'll start to be able to identify not just some of the rare Mendelian disease seeming alleles that might lead to that particular picture, but also the ones that cover the rest of the genetic landscape. Even without a molecular diagnosis, we can still treat our patients. And I think that's super, super, super important to remember, okay? Because I think as geneticists, we always stress about this. We're like, oh my God, how have we not diagnosed our patient yet, right? It's okay. It's just one assay. There's a million other assays, especially really good immune studies, right, that we can use to characterize our patient's immunobiology. And if your patient has a specific pattern, guess what? You can still treat them with maybe some of the drugs that work for other diseases with that particular immunobiology and get them well while you work on their diagnosis. That's one of the luxuries of working in the genetically driven immune disease realm, is that sometimes you treat them first and then you diagnose them. And if anything, their responses to treatment becomes another really important source of information about where you should be looking mechanistically for the answers for the diagnosis, okay? Okay, so basically this is kind of where we are at right now, right? Like we do this sometimes, I think, without even thinking about it, but it's important to articulate this for our colleagues who may not be as molecularly minded, right? So what we're doing on a daily basis when we are formulating diagnostic and management strategies for our patients is we're taking concepts that we know about clinical and genetic paradigms, right? And then we're putting it together with the tools that are at our disposal and what we know about their pros and their limitations. And we're essentially like synthesizing to come up with a strategy that makes logical sense. I'm going to talk very briefly. I have a bunch of slides, but I'm going to skip over some. I just want to give you like a quick snapshot, I think, of like how we clinically reason our way towards that particular diagnostic strategy. And then I want to spend a little bit of time actually talking about how our current thinking about our molecular assays and analyses are evolving. Okay. So clinical paradigms, of course, it's very important to know this if you're going to pick the right test, quote unquote, right? Really important to remember, right? B cells are kind of like a canary in a coal mine for something going on in your body, right? So when you have B cell issues, your patient might have problems with something that's B cell intrinsic, right? I mean, we call this growing pains, the Bildungsroman, this might be, a problem with a developmental gene regulation gene like Icarus. You could have B cell problems because you have growing pains, because basically the whole system of trying to grow up doesn't work, right? So these are like some of our genome maintenance genes. You could have problems because you have food insecurity, right? You're not getting the right nutrients, or you can't metabolize your nutrients properly. This is, disorders like congenital disorders of glycosylation or Golgi trafficking problems or many other kinds of inborn errors of metabolism. You could have B cell problems because you hang out with the wrong crew of people. You have T cell dysfunction, right? You're getting the wrong instructions is not your fault, but your friends have just gone bad and, they're telling you to do the wrong things. And you could also have B cell problems because you grew up in a bad neighborhood or your house is on fire and you're not living a great life because of that, right? So these are all important things to think about when you see that a person has B cell problems. Don't just jump to the conclusion that it's choice number one, right? So this is why it's important to think about all of the different kinds of processes you might need to begin interrogating that person for when you interview them about their history, when you do their immunophenotyping. And these are some of the broad categories we often think about when we're thinking about sort of, the kinds of buckets of diseases we should be barking up for, that patient in front of you with CVID, okay? Now, I usually frame sort of my pathobiological formulation in this way, and this is kind of like, in some ways, a very pseudo mock-up because many of these disorders fit into many of these particular zones very easily, but it's just to show you, geneticists are trained to be dysmorphologists, right? Like we match like, oh, the person has hypertelorism, does this disease have hypertelorism? You can't do that with immune disease. There's so much overlap. And so you have to think about matching pathobiologies. And the way you get at pathobiologies, the way you get at what are the primary mechanistic drivers of the disease, not the passenger effects, is by thinking about where, you know, those mechanisms might land a person on this map of the different facets of, you know, manifestations of immune disease. Certainly when I think about, you know, people ask me, what's your differential for CVID? Which gene panel do you send? And I'm like, no. If you don't know anything about the person, you're starting from the differential of all genetically driven blood and immune disease and going down, you know, narrowing down from there as you collect more data, right? You're not starting from a gene panel. You're starting from the understanding of like, this could be any genetically driven immune disease until I know more. And so within that bucket, you really have to think, okay, is this one of those diseases that's as rare can be? Is it one that's maybe individually rare but collectively common, where there's like, you know, a bucket of shared mechanisms, endotypes, whatever? Or is it one of those that there's like super common and, you know, kind of, you know, base some of the further assumptions you're going to have from there. I don't even have inborn errors of immunity and other diseases on here, as you can see, and I probably should, right? But in terms of the clinical, like, how do you narrow it down from there? Well, you know, some basic things, right? Like, of course, immunodeficiency. We all think about immunodeficiency. The person clearly has some kind of immunodeficiency, but then you sort of have to sit there and ask yourself, well, what kind, right? Because I think everyone knows how to screen for high penetrance, broad susceptibility to lots of infections. But you really have to think about, you know, could this also be a person who has, you know, high penetrance, narrow susceptibility to fewer infections? Could this be low penetrance, narrow susceptibility to few infections? I can tell you, I had a patient who had CVID because of herpes viral encephalitis. That person had such a bad post-infectious diathesis just because their body got like so messed up from the actual herpes virus encephalitis itself that their B cells were just like shocked into stagnation for a while. It took them like 1 to 2 years to like respond, and you know, and they didn't actually really need anything to help the B cells come back. It was just like the body had to stop being on fire slowly, and then the B cells started to come back. So, you know, never assume it can't be one of these other pathways or one of these other buckets. Okay. Other groups of diseases, I think this is pretty obvious, right? You have to think about primary adaptive immune dysregulation, of course. A-to-B is an interesting one because A-to-B can be quite complicated, and we always have to get to the bottom of, like, are we thinking about, is this like, you know, Th2 cytokine-driven inflammation? Is this person having some primary eosinophilic issues? Is this person having, you know, other kinds of like mast cell types of issues, you know, like that really kind of you have to make sure you're in the right corner with A to B. And then certainly with auto and hyperinflammation, a lot of times if you're treating your patient with a lot of adaptive immune agents and you're not getting any kind of treatment response or traction, you might need to think, are you treating the wrong mechanisms, right? Is it actually that your humoral problems are coming from these deeper underlying cell intrinsic or innate immune problems? So this is really just to sort of belabor the point, but, you know, our B cells grow up in bone marrow. This is like the cradle for B cells. So a lot of these patients can also have hematologic abnormalities, and that's another really, really good clue. And when you look at hematologic abnormalities, you again have to remind yourself, you know, it may not be bone marrow intrinsic. You have to remember that there are all these other potential mechanisms that you should consider if you see that this is a patient that has, you know, for example, cytopenias or even aplastic anemia. Malignancy is the last one, and it's really important to highlight because I would argue that almost all of genetically driven immune disease can be considered a pre-malignant state. And that's because if you think about the mechanisms by which people end up developing these conditions and what these genes represent, it's all of these same processes that are shared with tumorigenesis mechanisms. So your patient's first presentation of their disease may be cancer, okay? And then that becomes quite complicated to unravel because, you know, there's the question always of, you know, is this paraneoplastic or not, or do they have an underlying germline condition you need to worry about? The other thing is, we do not do a great job as a community of trying to understand the underlying immunoheme findings of various kinds of genetic disorders or syndromes that were discovered by the genetics community rather than the immune disease community. And we all have to help ourselves do this, because a lot of times when you're sitting there and you have what looks like a pathogenic variant and you're like, oh my God, there are no immune features described for this disease, you really have to think, is this truly absent, or is it just simply not examined for? And a lot of times, unfortunately, it's the second. I think, my friend Dushan's work on Down syndrome is a really great example of how to go about really thoroughly characterizing all the complex disease mechanisms for a condition. And he's done this for Down syndrome. Others have done it for, many other conditions, or starting to do it for many other conditions, such as Kabuki syndrome. But it's just really, really important to begin to sort of broaden our scope and try to understand better what pre-existing known genetic syndromes might do to immunity. So genetic paradigms wise, I don't think I need to teach this audience that, when we think about genetic testing strategy, we have to consider all of these different factors, and then we have to sort of put it all together. But, we deal with a lot of diseases that are really annoying, right? Because they have so much variability and they have so much incomplete penetrance. And that isn't always expressed just on the organismal level. Sometimes we have to deal with it, penetrance existing on the cellular, but not the organismal level. We have to think about penetrance in terms of overall disease versus specific features versus the timing of emergence versus responsiveness to certain therapies. So, how do you encode all of these different dimensions of complexity? That's something that we have to really work together towards thinking about. And all of these kinds of complexity, all of this whole, you can't just assume because you have a genotype, what your patient's specific expression of disease is, comes from these different layers of heterogeneity that my colleagues are going to talk about in more detail. But, we have so many different kinds of allelic mechanisms, not just per disease, but also per gene, and also sometimes per allele. And so when you're sitting there with a VUS, you have to really think hard, what are the functional studies I'm going to use to try to validate this VUS? It might be more than one. I might need, multiple different ones to fully understand the impact of my particular condition. And this is because of so many different functional tools we have, but also so many different ways in which those different assays might be modified on different levels. Certainly on the DNA level, we have to worry about a lot of ways in which, modifier loci in other places can affect both disease. It can affect disease expression on both the allelic and or the pathway level. We have to worry about somatic mutations, of course. We also have to remember that it's not just about, modifiers that are, HLA loci or things like that. Like, as human beings, we have basically a lot of places that were selected for in our modern human ancestors, because back in the day, if you were a person who could mount a slightly higher fever slightly faster, guess what? You were the one who survived and passed on your genes in the era where there was no hygiene and there was no antibiotics and there was no pasteurization. So those variants that once gave us all of this lovely resistance to life-threatening infection are kind of useless to us now, but they certainly predispose us as a human entity to inflammatory and autoimmune conditions. And it's really important to remind ourselves about this, because it doesn't always have to be rare Mendelian disease that's contributing to broken immune tolerance. Somatic mosaicism is really important, and certainly there's sort of two main buckets that we deal with, right? So we deal with a lot of diseases where the mutations confer a fitness advantage to a specific lineage, and then you get these like immune dysregulatory and inflammatory disorders. And you also deal with mutations that rescue a fitness deficit from, say, a ribosomal biogenesis disorder or bone marrow failure syndrome, which all also have, immune manifestations. And so you also have to worry about sampling the correct tissue to be able to get at that question of like which one. You have to worry about using different strategies to be able to detect these mutations. And this is incredibly important because you need this information to be able to provide proper counseling to your patient and their family members and know what additional risks they need to be assessed for, like malignancy. I'm not going to talk too much about RNA level changes. Of course, I think everyone has heard, a lot of the recent really interesting stories about random monoallelic expression and stuff like that, where, we have to worry about transcriptotype, quote unquote, rather than genotype that might determine intrafamilial variability. There's certainly lots of proteins that can modify, phenocopy or rescue GBIs. And, there's people at the NIH who study this and colleagues of mine like Mike Abers. And that's definitely something that we don't have great tests for necessarily for all of these different kinds of autoantibodies floating around. And then, of course, we have to apply this to every patient's specific context, which means a lot of, logistical considerations as well. But when we do this, you end up with this ideal situation where, you go from the clinical evaluation all the way through post-test counseling or reverse phenotyping. But most of us actually get stuck in this particular phase of variant triage and classification, right? The dreaded VUS. And so right now, at least in the US, I tell my patients that probably no more than 3 to 4 out of 10 patients that have strong suspicion for genetically driven immune disease are going to get an actual diagnosis despite the fancy clinical grade testing that we're sending on them. And this is not just because of limitations of technology, which are probably the easiest ones for us to deal with. It's also because of these various different kinds of limitations of knowledge, beginning with the fact that we're still shit at phenotyping, right? So, phenotyping is a garbage, when we do genetic analysis, it's a garbage in, garbage out proposition, right? It's only as good as the phenotyping provided, and we're still just simply encoding things in HPO terms for the most part when immune disease is so temporally dynamic, and we don't really have a great way of expressing cause and effect. We also have not very good reference diversity and representation. I have a lot of patients in the Bronx with ancestries that are not going to make it even close to some of our reference sequences or our population databases, right? We have a lot of assumptions about genetic architecture that are probably wrong, and we don't even know it. We have databases that can't keep up with the pace of discovery, and certainly we have problems with our variant classification framework, which is kind of the next thing I want to talk about briefly and end on. So, we work in a disease domain where we have more VUSs than any of these other disease domains, despite the wealth of tests that we have at our disposal to help us resolve VUSs. And that's always kind of interesting, right? Why is this so true? Well, certainly part of it is not our fault, right? We can blame part of this on the ACMG. I'm not even joking. So, the ACMG 2015 classification framework, which really, people are calling variant interpretation frameworks now because classification is a terrible term. This was largely predicated on assumptions from classic Mendelian disorders, aka non-immune disorders, where we mostly just understood the paradigms related to small nucleotide variation encoding genes, okay? This is not the wave of the future. They left it vague, because they recognized that. The problem is that even with the vagueness, even recognizing that you need to leave room for subjectivity and domain-specific paradigms, a lot of the genetic assumptions do not work with genetically driven blood and immune disease, and I'll give you some examples. Certainly, some of the structure of the current variant classification or variant interpretation system also needs to be changed, and that's true for all diseases. And this is actually happening soon, hopefully in SVC version 4, which is I'm told going to come out by the end of this year. Some of those lines of evidence that you guys who do a lot of variant interpretation are probably really annoyed by, hopefully will go away because a lot of the double counting you've noticed, a lot of the non-independent weight contributions, a lot of the evidence concepts that don't seem to make any sense and maybe conflict with each other, those are being restructured to be less annoying. So that's that's great news. Okay, but some of our problems in our particular disease space may not be as easily solved, okay? One of these is, we have to deal with a lot of incomplete penetrance and variable expressivity. So a lot of these de novo rules oftentimes are tricky to apply, right? Sometimes we just throw them out. We have genes where there's multiple gene-disease relationships we have to classify for, and a lot of those mechanisms don't fit the simple PVS1. If it's null, you get this, and if it's truncating, then it's this, and if it's, in-frame exon skipping, then you're done. No, right? It might be a whole other disease. Other problems. This is one really big one, and I actually have no idea how we're going to reconcile this over time. But it's this concept for, most geneticists would tell you they don't consider a functional study to be anything done in a patient cell, right? Because you have to take that variant out of the context of the patient's other 23,000 genes in order to really prove pathogenicity. Of course, for us, the problem is like, well, how biologically relevant is putting, a gene that's expressed in CD4 T cells into a breast cancer line, right? Or are there mechanisms like destabilization of protein that you're not going to see through overexpression in vitro? There's a lot of amazing patient cell-based assays. People are developing more and more patient cell-based assays to try to get at functionality. And yet, if we're not being allowed to actually use these as functional criteria, we've basically screwed ourselves, right? These are basically still being considered cellular phenotypes right now, and they're getting lower weights. We have so many other problems that I'm not even going to go into, but suffice it to say, right now, there needs to be more of a voice from the immune disease community in terms of how variant interpretation occurs, because otherwise we're not going to have the genetic paradigms that we deal with represented in even the way we make rules. We're going to be hamstrung, okay? So I encourage everyone to, join ClinGen and, look out for SVC version 4, which hopefully will, do all these lovely things to revise our current ambiguities. And I'm going to skip this part. You don't need to know these details, but talk to me afterwards if you're interested, of course. There's a lot of stuff going on as well in terms of trying to address, the non-coding, non-simple germline single nucleotide variation types of mechanisms, right? So there's groups now thinking about how are we going to come up with guidelines for interpreting copy number and dosage variation and somatic variation and, something that is not a very, high penetrance, high variant effect kind of variant. So, those are all very, very useful things. But as a community, as a global community, we still have to do something about 1.7 million VUSs, right? And how do we do that? Well, we have to figure out, who's going to functionally test all of these with, hopefully minimum of redundancy, who's going to develop and perform the assays that are needed that we don't have for some of the gene disease relationships, who's going to store all of this stuff, who is going to decide how the data is going to be made available and to whom, who's going to provide the regulatory framework, who's going to provide the actual data infrastructure, who's going to, make sure the data is appropriately shared, standardized, and harmonized, who's going to make sure we properly incorporate that evidence into our current variant interpretation schemes, and who's going to pay for all of this, right? So these are all unanswered questions that the international community needs to come together to address. And with that, I just want to say, never forget that, this is an ongoing journey with our patients. So, don't get frustrated. We always get frustrated because we can't get an answer on the first pass genome or whatever. Walk with your patients. They'll teach you more about, their disease over time, and you'll get there. So hopefully I've just imparted some very basic lessons that genetically driven immune disease has taught me, certainly, and I will stop because I'm probably over time. Last message, immune disease evolves over time, our genomes evolve over time, so our thinking certainly should continue to evolve about our patients. Thanks.
Thank you, Xiao. It was really a great talk, really educational, and the broad view of genetics in AI or GBI. I don't know if there is some question in the room, or we can check. If you want to do your question in the Zoom, you can put it online, and we'll ask Xiao your question. If not, meanwhile, I can start maybe with a few. So my first question is, in your patient, when you stop looking for genetic results, so you have like no, at what I know that it's never, but when you start like obsessing the search for a genetic result?
Yeah, it's actually a pretty simple question, I think, in some ways, because we all have limited resources, right? So, I the sicker the patient is, the harder I look and the faster I try to work. And this is why, I'm really, really lucky to have the use of non-genomic tools as well, because a lot of times I end up doing that thing I was talking about where I try to figure out what the right medication to stabilize their disease condition is first. And then that buys me time to get them to diagnosis. So the goal, of course, is always to make your patient better, right? So I stop, or I slow down, I should say. I don't stop, but I slow down looking when the patient is in a really good place. And I stop when the patient asks me to stop.
Okay, thank you, Xiao.
That's a good one. Thanks a lot, Xiao, for being here in early morning. We really appreciate you, and it was a very nice educational talk. I have a couple of questions/comments. The first one is about the syndromes where may have immune manifestations that we missed or overlooked, because we're working on that also. And in fact, in some syndromes, there are clearly some increased infections or autoinflammatory things that you can pick up. But in others, we found out that there are subclinical things that haven't been studied yet. So the barrier here, at least in our case, was that these patients are usually followed up by neurologists and so many other doctors, but usually immunology is not involved on that. So it's kind of difficult to approach. So how do you think we should go? Just a bit more immunology training with the others, like multidisciplinary meetings, or start with the syndromes that do have immune manifestations and then move to the others?
Yeah. So that's such a really good point, because I have the luxury of being a geneticist too, and so of course, like usually this gets found for me in the context of a person who actually was sent because of concern for existing immune problems already, right? And of course, a lot of times my problem is trying to figure out if it's because they are hypotonic as well and aspirating all the time or something. I would say probably the most important thing is what you just highlighted, which is education. So if you, I think a lot of times, the reason why genetics doctors, at least in the US, don't really think about immune manifestations of disease, even though they're usually very, very thorough about many of the other, kinds of specialties and organ tissue, whatever domains, is because it's not something they ever see in their training. Geneticists have zero questions about immune disease on their genetics boards. If they're lucky, they see one patient with immune disease, like during the entire course of their training. And so and part of it is also because immunologists have been so good at doing their own genetics that there's not enough crosstalk between the two clinical domains. So it's really, really important, I think, to teach our genetics friends. And actually, this is another reason why some of my colleagues and I, want to try to also write more literature, write more reviews about even just, the manifestations, the, immunoheme manifestations of syndromes that we currently know about, because a lot of those things that are even, already in the literature, people might not know. So it's really, really important to educate, and it's really important to reach out and, form collaborations and be like, hey, this particular gene that you for this particular disease, in these patients you follow in epigenetics clinic or whatever, they're really well known to cause all these problems with blah, 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 blah. Do you have any patients who have any of these manifestations? Maybe if you do, you could send them to us so that we can make sure that they're well taken care of from that perspective. And sometimes, it can be something as simple as boiling it down to like this very short review of systems. Like if your patient has, A, B, or C problems, send them to us. And that really, I think begins a very good collaborative dialogue.
Thank you. So there is a couple of questions online. The first one, I think it's a bit a controversial question. So do you think, Xiao, do you consider that CVID, when there is a variant, a pathogenic variant found in a gene with a phenotype of CVID, if it's still CVID or if it's then another thing?
So I have this conversation with my patients a lot, and it's not just about CVID. So I'll tell you what I tell them. I usually say, look, you're probably going to end up with a lot of different kinds of clinical labels, okay? And you can have as many or as few as you want. I don't care, whatever makes you and your other doctors happy. But just so you know, the only one I care about for you in terms of treating you is the reason you have your problems, which is that particular, molecular genetic diagnosis. So I will speak from that level of organization, but it's absolutely fine to have different kinds of clinical labels, CVID or others, associated with that particular molecular diagnosis if it helps, for example, you to get this medication from insurance that we need from you, or if it helps this particular doctor better communicate about their conception of your disease in a way that makes sense for them. But let's just not be confused about what's the cause and what is sort of the description.
Thank you.
Yeah, I think that maybe we can save the other question for the roundtable because we're already a bit late. So thanks a lot, Xiao. That was wonderful. Also, a lot of people congratulating you in the chat. And yeah, we'll see you at the roundtable for a bit of an extended debate. Thank you. Okay, so after this super nice overview, we are moving to the first block, which is going to be focused on genetic mechanisms underlying CVID. And for that, it's my pleasure to introduce Dr. Andrés Caballero. Andrés is a postdoctoral researcher at the University Hospital Freiburg and the satellite center of the RESIST Cluster of Excellence at Hanover Medical School. His works exhibit on the intersection of immunogenetics, bioinformatics, and translational diagnosis. And he's also working on building platforms that integrate all these things to try to advance in research and medical care. So the floor is yours. Thank you, Andrés.
Thank you, Laura. Thank you personally and all the organizers for thinking of me and inviting me to this session today. I'm very happy to be here. And I would also like to personally thank Xiao P, my dear colleague. I mean, we've been working together for the last 5 years, or more than 5 years. And it's always such a humbling and learning experience to listen to her talks. And this is why you will see a couple of slides that we share because of the work that we've been doing together in recent years. So today I was asked to speak about this, the role of TACI in antibody deficiency or CVID. And so I will start by telling you, but probably you already know what TACI is, or TACI. So it's a transmembrane receptor expressed mainly on the surface of B cells, including, where can I point? Is this? No.
I think you have to point there and it goes there, or maybe not.
Or not. Okay, sorry. So then, well, you'll see it's expressed mainly on the surface of mature B cells, such as, so I continue? Okay, so class switch memory B cells, marginal small B cells, and plasma cells. So it binds to APRIL and BAF with similar affinity. And it is essential for T-cell dependent, but not for T-cell dependent responses. So it supports T-cell dependent responses, but it's not required. So once the ligands bind to TACI, so it's the molecular signaling occurs and it induces NF-kappa activation or NPAD activation, and this promotes class switch recombination, antibody production, plasma cell differentiation, and plasma cell survival. It also takes care of maintaining this long-lived antibody responses. So it's also important in general for B-cell homeostasis and tolerance. because it takes care of controlling the size of the marginal zone basal and defollicularized helper compartments, and avoids that there's an excessive production of antibodies and expansion. Ensuring the tolerance of the system. So why are we interested in TACI or TACI? So what is its role in disease? So it has been known since the year 2025 when it was first reported that. Okay. So since it was first reported, it seems that it has been found to be present. So let's say rare variants in this gene have been found to be present in about 1/4 of patients that have been put the label, as Xiao P said, of CVID, for whatever that means, right? And so this tells us that, I mean, it does have a role in disease, and we want to understand better why. Because the thing with these rare variants in TACI is that they're also present in the general population. So TACI, or these variants in TACI have not only been associated with CVID, but with selective IgA deficiency, and with predominantly antibody deficiency, and also with autoimmunity. So it's important to note that both biallelic and monoallelic variants are associated with disease. And surprisingly, the biallelic variants they cause a less severe phenotype, but they are more penetrant. So that means that anyone carrying two variants, so both chromosomes of the gene, will likely develop a disease, whereas those with monoallelic variants are less likely to develop disease. So as you can see, I was speaking about the low frequency, allele frequency of these variants in the population. So what we think of control or general population, I'm just showing you here a table of rare variants that we find in our cohort of patients that we see in Freiburg in Germany. And just together with the allele frequency, so you can see that for each of these variants, you can see the allele counts, allele numbers. So each of these variants, they can be present in about less than 1 in 1,000 alleles, except for two variants that are quite relatively common. So the C104R and the A181E. So those two, each of them can be found in 0.5% of alleles in the general population. So this is data taken from NOMAD V4. So and now, this is some work that we did, what we published in 2021, just summarizing the prevalence or the frequency of these variants in our internal cohort of patients that came to our attention in Freiburg, that they were screened with targeted gene panels. So we did not do whole exome sequencing, but just a targeted gene panel of about 200 or 300 genes. So you see that each of these variants, if you compare them with the controls taken from the NOMAD database or internal controls, so taken together, it's about 2 or at least 2% of individuals or control individuals will present one of these rare variants. And if we compare that with the antibody deficient patients, so being with CVID or predominantly antibody deficiency, so they're present in about 10% of them. So there is clearly an enrichment of these variants in the antibody deficient people. And if you look at these two more common variants individually, so they are about 3 to 3.5%. So this is not allele frequency, is individual frequency or subject frequency. So 3 to 4 people out of 10 carry one of these variants, and taken together, so about 7% of them would carry any of these two. Just wanted to update. So a similar analysis, a more recent analysis, taking not only individuals that have been sequenced with targeted gene panel, but some other subjects that we have sequenced lately with whole exome sequencing. So you can see that the numbers go a bit higher. So if you see the controls, or the patients, instead of 10%, the percentage of individuals, and this includes not only patients with antibody deficiency, but with other immune-related disorders. So the percentage of individuals that have any of these TACI variants is about 13% or more. And individually, the 104R or the A181E, so that together they also go up to 7 or 8% together. So now if we focus on not taking the whole cohort of individuals with immune deficiencies or other immune-related problems, we see that when we select the predominantly antibody deficiency or CVID cohort, so about 10% of them would carry one of these TACI variants. And if we look at all the TACI carriers in our database, so we see that about 73% of them, they're likely to be tagged as affected or patients requiring treatment, whereas still there's a small proportion of unaffected or unknown, probably because they're family members or internal controls. But there seems to be relatively high penetrance, when we compare with, as opposed to what we might think of these variants before. So it's a similar penetrance to that we see for CTLA-4 insufficiency or NF-κB1 insufficiency. And just going back to that study that we reported in 2021 on subjects with predominantly antibody deficiency sequenced or screened with targeted gene panels. So we see that the proportion out of those cases that we consider molecularly diagnosed or solved or possibly solved, about 29% of them, they could be explained or partially explained for variants, for the variants that we found in this gene, which is similar to what we found in that other review that shows that about 25% of patients that have been labeled with CVID would be expected to have one of these variants. So now going to the article that I was asked to speak about here today. So this is a work that we did in collaboration with different groups from the US, Iran, and Sweden. Because of this, to try to solve the mystery of why still there are so many individuals, control individuals or general population that carry these variants that do not develop disease, and why there are some that they do develop it. So what are those additional factors that are contributing to disease development? So the study, this started 2 years ago, but it was published last year. So we collected from these 3 different centers, we collected a total of 161 individuals, so symptomatic, predominantly antibody deficient patients. So the rule of inclusion was that they would present one of these rare variants in TACI. When I mean rare, it would be less than one frequent, with a frequency of less than 1%, and with a predicted high or moderate impact. So that means that it would that variant would be a missense change or a stop codon or a frame shift or a splice site. So that was the inclusion criteria. And so we collected, and we needed to have whole exome sequencing data because we wanted to look for additional hidden factors, genetic factors that could predispose or exacerbate the presentation of the phenotype. So this table just gives you an overview of summarizes the clinical features, demographics of these individuals. So what is remarkable from here is that all of them, 100% of them presented with recurrent upper respiratory tract infections. About 80% of them, they were put the label or diagnosed with CVID. And one of the significant differences about this cohort is that when we compare the monoallelic and biallelic patients, so there was a small or significant difference in those patients that presented autoimmunity, which was more prevalent in the monoallelic ones, and the presentation of bronchitis or sinusitis in those with biallelic variants. So here's this, again, the overview that you can go and find in the paper, an overview of the immunologic profile. So reduced levels of B cells, immunoglobulins, and the most significant difference that you can find when you look at the monoallelic versus the biallelic patients is that the patients with IgA. So if you look at IgA, it's more reduced. I cannot see the table now. So 15 to 27.5%. So the biallelic is more reduced than the monoallelic. So in this cohort, we found 38 distinct variants across the whole cohort. So you can see the percentages there. So almost 90% of them, they were monoallelic, and about 12% they were biallelic. So we found both compound heterozygous situations and homozygous. And again, the most common variants as expected, they were the C104R and the A181E, along with this frame seed occurring from the 16th amino acid position onwards. And there were only just, as a curiosity, only 7 variants that were present in compound heterozygosity, so never found as monoallelic. Just here to sum up, when we look at this cohort, the patients with biallelic variants, they have a later disease onset. As I said before, so it seems to be that they are less severely affected. So that's why they also have a longer diagnostic later onset, longer diagnostic delay, or they also presented later in the study, higher rates of sinusitis and bronchitis, as I said before, and they are more likely to have lymphoproliferative disorders. And again, so the IgE levels that I was mentioning before. So when we look at the cohort, depending on the variant type, so patients with non-missense mutations had a higher susceptibility to cancer, and those patients that had variants localized in the extracellular domain had a higher frequency of enteropathy as compared to those in the intercellular domain. But now going back, so just here to give you a brief overview of the amount of genetic variation that we find in this population. So this was one of the first questions. So is it one of the reasons is that a second hit in another gene, let's call it an immune relevant gene or IEI gene. So we see that in this population, so every bar represents one of these 161 individuals, and you can see the number of variants. So when you do whole exome, so after the whole exome sequencing analysis and filtering, you would come up with about a range between 5 and 50 variants per individual. So rare candidate, possibly important variants in other IEI gene. If you narrow down that to the variants that fit the mode of inheritance or the reported modes of inheritance at that point for these genes, so the numbers go down. So with the maximums, with even one subject or two reaching 15 candidate variants, but others that have even none. And there are, so if we go one step further and we try to apply the ACMG guidelines or criteria to classify variants as pathogenic, or I mean, as Xiao P perfectly mentioned, so they really, I mean, it's difficult to apply them in our disease realm, right? But even with that, so it seems that at least 20 of these individuals would carry variants that fit the mode of inheritance, and they would be classified as pathogenic and thus disease relevant. So it could be that these patients had another diagnosis. It could be that actually it's a combined effect of the TASI variant and this other variant in the second gene. So if we then compare this cohort of 161 individuals that we selected with another internal cohort of antibody deficient patients taken from the Karolinska Institute in Sweden, we wanted to see what was the enrichment, if there was or not an enrichment of these rare variants in particular genes in our selected cohort compared to that unsolved cohort. And we saw that there were 3 genes that would pop up or become more significant. So individuals with variants in TASI would also have variants in DKC1, GACN1, JAK1, and CXCR4. So those were the more significant differences. It doesn't mean that they're necessarily mean that could be pathogenic or they're making an influence, but this is what we observed. And if we do the same exercise, but looking outside the list of genes that we have or that we know as IEI genes. So in any other gene in the genome, applying the same type of filtering, so rare, like below 1% allele frequency, and high and moderate impact. So we could see if there's an enrichment of certain variants that are present in our population, in our study cohort versus the other cohort. And so it seems that, of course, there were some variants or genes with variants, right, coming up, but there was no specific signaling pathway enriched. But there were some genes that are actually involved in visual function, or that are important for visual function and plasma cell development, so as those listed there. And so finally, the third approach was to look for not so rare variants, but maybe something more common, like single nucleotide polymorphisms. So type of GWAS analysis that we did was basically to compare, again, our cohort, our TACI cohort, with the other predominantly antibody deficiency cohort, and see if there were certain SNPs that they were more enriched in one cohort versus the other. And as you can see, yeah, there were some popping up with more or less significance, and those genes, they were involved in the mismatch repair pathway, PIK3 kinase pathway, and so on. And you can see how those genes have been linked by gene ontology with certain things like lymphoma, laryngitis, and so on. So finally, we tested whether there was a link between, or an enrichment of certain HLA types in our TACI population versus the other population. And we found no significant differences with the HLA class 1 alleles. But we did find some differences for certain HLA types and HLA2 types. So DPA1, DVR1. So you can see those that the first one was exclusively expressed in TACI patients, and the second one was almost absent in TACI patients. And there were other alleles that you can read there. So there were also significant associations with this TACI cohort. So in summary, if we take together, so depending on whether we want to take together those 20 cases that had other possibly diagnostic findings, it would be that about 50% of the population still would not have an explanation why they have disease. I mean, only with the TACI variant alone. But there were a small proportion of individuals that could maybe be explained by additional factors, such as a second allelic variant in TACI, an HLA type, a specific HLA type, a specific SNP. And again, if you don't take those individuals into account, up to 60%, still sadly, 60% of those patients would maybe remain a mystery of why they develop disease. So I'd like to thank especially Hassan Abul-Hassani, who was the main leader and person who took care of this work. All the people, so Michele and Bodo in Freiburg, and the colleagues in the US, and of course, so Samane and Nima in Iran. And thank you all for your attention.
Thank you, Andres. It was very nice. And I think that this is a very hot topic right now, because especially as a more geneticist perspective, we had a question in the chat about how we should report different variants. And I think that for TACI is one of these special cases that sometimes people doesn't really know how to interpret that, because as a geneticist, you sometimes report a BUS, oh yeah, it's high frequency, whatever, but then people has these genetic results and need to move forward, especially for genetic counseling. So you have shown us that in some cases there's a second hit. I think that the general recommendation is that look for that for another rare variant, but for the patients that only carry one monoallelic variant but do have specifically common viral immunodeficiency phenotype, what would you recommend in terms of reporting? Difficult question, sorry.
Yeah, I guess it's a question that everyone would ask when it comes to this gene. Yeah, I mean, still such a struggle how to deal with the putting the right label to these variants. So, and of course, I mean, I myself, I would not know whether you should call it pathogenic, risk allele. I mean, what do we do with this? I mean, according to the criteria, I mean, it clearly has a role in disease. I mean, we've seen that, and we keep seeing that because it keeps coming up. So, you know, every that 10% of your patients with antibody deficiency will present one of these variants. So it's clearly enriched. It's clearly doing something. I mean, there's a molecular defect, I mean, that is shown. So, but of course, how do you explain that? Yeah, some other people do not present any symptoms, right? Which I guess we have to get used to that because it's not only happening now with TACI. I mean, it happens with CTLA-4 and with others. And so what I would suggest is, yeah, maybe if you have the resources and the time, yeah, maybe you want to keep investigating and not only stay with that diagnosis of, I mean, with the variant that you find in TACI, but maybe you want to, you know, do HLA typing, which may be something simple to do for some labs. Of course, look for additional findings. I mean, don't stop at, you know, if you find that variant in TACI, don't stop there when you're doing exome analysis. I mean, try to find a second hit. Yeah, and of course, I mean, there are other things that you could do, explore. I mean, there's, of course, epigenetics, microbiome. So, but of course, it depends on resources and time. Yeah.
Yeah, great. I don't know if anyone in the audience, Roger, you have a question? Yeah. And then we have a question online.
Hi, Andres. Thank you for your talk. It's a very interesting study. So my question is, you show a list with all the TACI variants that you found in your cohort. So my question, and linking with the question of if we should report or not TACI variants, is that besides C104R variants, which are these and the 181, which are pretty demonstrated that are deleterious and functional demonstrated, what about the other variants? So you include all rare variants that you obtain, or you filter for variants that are demonstrated to be deleterious?
Yeah, I think that probably the issue with TACI is that I guess it's not so exciting anymore. You know, I mean, this was first discovered in 2005, and now it feels, I have the feeling that it's not so cool, not so, and people probably stopped functionally testing them, right? So they just, you know, we know that these two are the most common ones, and the other ones, yeah, maybe do something similar, but I guess everyone assumes that this gene is already solved, and which is a bit sad because I think that there should be more testing done. I don't know. So someone, I guess, it would be nice if a lab would take the lead and test all these variants. Because I mean, there's clearly an effect. I mean, it may not explain 100%, but of course, I think, I mean, not all of those variants are tested, but I'm sure that, I don't know, I could say that 80% of them are relevant. You know, there's also this I87N, so that's also, I mean, shown to be is not so common, but I know that there's also some functional proof. So, but yeah, I think that's, we need to test. I mean, I don't know, the community needs to go back to TACI, I think, and test.
Yeah, I fully agree. And so as a community, we would appreciate if anybody can
takes the lead and
can update the functional studies, because when I inform some of these variants in my genetic reports, usually I cite papers that are quite old. Exactly. This does not mean that these papers are pretty good, but probably it should be worth to test it again, not all the classical TACI variants, but the other ones comprehensively to update all this functional knowledge.
Yeah, exactly. I mean, and I mean, mostly now when we have the tools, right? I mean, there are so many genes now that are being studied as this MAVE system, right? So I think this should be the way to do this. And of course,
yeah, absolutely. I also agree. We have one quick question in the chat, and is if you have found any association of TACI variants with nephropathy phenotypes, specifically primarily glomerulonephritis. You have.
I mean, we don't check for that. So there might be correlation, but the thing is that, I mean, that depends on how the documentation of the clinical documentation of the patient happens, right? And I think at least for our, I mean, we don't know, maybe the patients have it, but if the doctor that who's referring the case does not mention that, we will never be able to test if there's, yeah.
Yeah, this links a bit with my question to Xiao, that sometimes in other syndromes, if we don't look for it, we don't find it. And finally, we have a comment by Bodo from afar, which is, why do we not just classify the TACI C104R as a risk allele? Cancer geneticists usually do this. Same true for other TACI variants.
Yeah, I mean, I think it's, yeah, so as I said, for me, the label is not so important, and it's still, I mean, conflicting, because, yeah, you know, if you follow the guidelines, you know, might be risk-allele, might be pathogenic, but I think what it's important is that it has relevance, and I guess not so much the label, and that I think what's important is that it should be reported, and that's, I mean, what I would stay with.
Yeah. Nice. Just a reminder for people online, we have more than 120 participants, and everyone can ask the question, and also for this open call that we were doing about functional validation. It would be super nice if someone knows of something, you just can put it in the chat and we'll discuss it during the roundtable.
And Laura, on that, Xiao made a comment before also. Just remind that ESID has a document where it is listed all the labs with the functional testing, at least in Europe, so it can be useful also for checking variants depending on the disease that you have.
Yeah, thank you, Andres.
Thank you.
Great. So we have one more talk before the coffee break, which is Professor Ferran Casals. Ferran is an associate professor in the Department of Genetics, Microbiology and Statistics at the University of Barcelona. He's also a member of several leading research institutions and some networks as CVID. And Ferran has a very nice background combining evolutionary genetic, population genetics, and also medical genomics. And today he will present the oligogenic model in CVID. The floor is yours, Ferran.
Okay, thank you very much.
It will appear, yeah.
Oh, I think you should go some slides before. Yes, more. Yes, this way, sorry. I understand.
They were confused.
I understand the confusion, so that's okay. Well, thanks to the writers for the invitation and all of you for your attendance. I don't know what you see here, okay, but this is maybe you see an optimistic message because it goes up, okay. But believe me that this is the main message and our main results I want to present today. And this is the idea that I want you to keep with today. If you understand the model, I will be happy. Then we will see results. But I would like you to understand the model and to challenge our model. Okay, so this is a model to test the dygenic inheritance in rare disorders and specifically in inborn errors of immunity. I will give you some hints. Okay. Well, now you see observe and expect, so maybe you are realizing we're going to talk about population genetics, okay? I have asked to close the doors by the outside because you don't run away. But all of you use population genetics, okay? So raise your hand if you have never filtered a variant because of the high frequency, for example. You have done this. This is population genetics because what you are doing is you are assuming that natural selection is keeping at low frequency the variants that have a harmful effect. So this is population genetics. Or another example, you don't need to raise your hand, but many of you, me, myself, we have the O blood group, for example, right? We have a loss of function in our genome in homozygosity, and we are apparently okay, right? Well, it has a high frequency, so we are pretty sure that it has not a harmful effect, okay? And this is the key idea, okay? The same way that a variant with a harmful effect cannot reach a high frequency, okay, because it's dangerous, okay, natural selection, it's keeping this at low frequency. We have the hypothesis that combinations of two different variants in two different genes cannot reach a high frequency if they are harmful, okay? So this is the idea, and this is what we want to test, okay? So let's put this aside and let's do some numbers. Imagine that we have two variants, okay, variant 1 and variant 2, that are quite common in the population, okay? You know very well that from the allelic frequencies, we can calculate very easily the genotypic frequencies, okay? There is a mistake, by the way, it should be Q in the last line. Again, this is population genetics, and you use this every day. In fact, when a variant is failing Hardy-Weinberg equilibrium, you discard this because we assume that there is a technological mistake and we discard this, but we use every day Hardy-Weinberg. Okay, so the same way we can calculate the genotype proportions from the allelic frequencies, we can calculate the probability that an individual is a co-carrier of two variants. Okay, being a co-carrier can be different scenarios. You can be homozygous for one, heterozygous for the other, but let's do it simple. And to have a statistical power, we have collapsed into 4 categories. You don't have any variant, you have the first one, not the second, the complementary, or you have the two variants in homozygosity, for example. Okay? So we can calculate the probability that an individual is a carrier, a co-carrier for the two variants, in this case in homozygosity. And if you multiply by the number of individuals that you have, and it is not arbitrary, this is the number of individuals that we have currently in the UK Biobank with genome data that is accessible. So you represent it here, okay, and then you go to the population and you count how many individuals in the population are co-carriers of these two variants, okay, and you compare, and this is what you get. You have a very nice line because, by the way, if you have pleasure doing these numbers, contact us because we may have an available position very soon for a PhD student, okay? I was forgetting this message, okay? So the point is that if you do this, it's incredible, and this is an idea that we have some years ago, and finally we have developed, and it's very nice to see how it fits perfectly, perhaps too perfectly, because what we are looking for is this, okay? Again, our hypothesis is that a digenic combination. Let's put, I understand it can be naive, but let's put in the simplest scenario, okay? We have two variants. Individually, they don't have an effect, but combined, they have an effect, okay? If they have this effect, you should agree with me that the observed number of individuals in the population should be less than the expected number, okay? This is the simple idea. Then you will see it can be more complicated, but this is the key point, okay? So now, and this, again, the results you will see in a while for CVID. And now this is the first slide. So my 15 minutes, they stop now, okay? I'm kidding. Well, so this is what I want to present today. We as a community, we have been for many years looking at rare disorders as monogenic disorders. This is okay. This is nice because in the last 10, 15 years, we have diagnosed thousands of patients. Depending on the disorder, it can be between 10, 20, or even more. 50% of patients of some rare disorders are now diagnosed. What we do in our lab is asking what happens with the rest, considering different models, somatic, allelic expression, structural variants, but also the oligogenic model, where a rare disorder, it's not generated by a single gene, but by a combination of few genes. It can be oligogenic, a few genes, digenic if it's only two, okay? Today, I will mostly focus in the digenic. If I have time, there is a slide for the oligogenic model, okay? So this is a model that we developed with Nerea some years ago, and the first publication, well, this is just to put some figures of how a digenic interaction can be. We have different models, different possible scenarios. Let's just stay with the one to the right, the left, as an example. We have two proteins that they form a dimer. The dimer keeps working with one mutation or the other mutation in the other gene, but it's not functional, for example, when we have mutations in the two genes. Okay, it can be also because they interact in a pathway or they interact in a more complex situation. Okay. At the genetic level, we could differentiate what is a classic digenic scenario where we have the example I have put, two individual variants that individually they are not harmful, but when they are combined, they can have an impact in the phenotype. Or the other situation, that is the modifier situation, where we have a variant that it's conditioning the expression of the other one, okay, or tuning or adjusting the expressivity of this second variant. So as I was saying, with Nerea, we developed this model, and the publication that we did, well, we have a model, but we have to test this. And then what we did is testing this with some diagnostic combinations that were already reported in the literature. Okay, and we went to DIDA. At this moment, it was called DIDA. You know that now DIDA is called OLIDA, okay, because it's now oligogenic. And what we did is testing the combinations in OLIDA. But we only test 5. Why? Well, because we need hundreds of thousands of individuals. Imagine you are testing the combination of one variant at 1% with another variant at 1%. The expectation is that you have one individual in 10,000. Okay, so to test that you have an excess or a lack of individuals compared to the expectation, okay, you need tens of thousands of individuals. At this moment, we were using genomics England data with 38,000 individual European individuals. So our analysis was restricted to situations where we had, well, I wouldn't say rare, or sometimes it's a combination of a rare with a more common variant. Okay, but among these 5 combinations that we were able to test, and again, it's very nice to see how the numbers fit perfectly. You expect 36, you have 36, okay? You expect 21, you have 20, okay? We are not saying these combinations are not true. We are just saying that they don't fit to our model, okay? But we had a couple of them, especially the last one, that was significant with a much less number of individuals, co-carrier individuals compared to the expectation, okay? It was the model that we did at this moment. Then we estimated how many individuals do we need to have more power, okay? And in the last figure of this paper, we report this figure where we have what we do is estimating the statistical power that we have depending on the number of individuals. In this case, we consider 500,000 individuals because we knew that UK Biobank was going to release this number of individuals, okay? And what you see here is that, well, we have, depending on the frequency, the higher the frequency, the more power you have to detect, okay? And also depending on the penetrance, okay? The predictions were quite high, okay? So with still medium penetrances, we predicted to have power to predict more diagnostic combinations using the UK Biobank data. And it came the day where we had available the UK Biobank data. And what we did, I will go to CVID in a couple of slides, but I want to present first the first analysis that we did, and also because this is where we are having more promising results, okay? So what we did is using, we tested 67 autoinflammatory disorder genes. And what we do is compare any SNP, any variant in one of these genes against all the variants in the other genes, okay? In this case, it was sort of a pilot project. We did only genes in different chromosomes. You will see that when we are in the same chromosomes, we have problems because of linkage disequilibrium, okay? So to simplify, we did only, so to avoid, to exclude completely the possibility that the results, the deviation, were because of linkage between positions, okay? So in this case, as you can see here, it was 67 genes. We're using genotyping data. So this is the total number of comparisons that we did. This is just to remember that we use the PCI values in the UK Biobank to only use European individuals, okay? If you use non-European individuals or if you use a mixed population, you can have also some results because of this, not because of a biological signal. And the results going directly, so again, so this is the first result, a straight line, very nice because observations fit to the expectations, okay? But when you do a zoom, so note that here, well, expected is 250,000, okay? So maybe if we are in rare disorders, we should look at the left part of the spectrum, okay? And if you do a zoom, sorry, well, you will see it there. If you do a zoom, so below 5,000 individuals, you find some points, and especially this one, that departs clearly from the rest, okay? It's not going down, it's going up. We will see now why it's happening, but you should agree with me that there is something clear, okay? Here we have the distribution of p-values. A problem that we have is that we are generating an incredible amount of data, and you will have very high p-values, but because the number of comparisons you are doing, it's very high, okay? But note that, well, most of them it's 1 because we have this very clear correlation, okay? But you have some very low p-values, but more or less they are following an expected tendency, but you have some increase here. Okay, what we say is that here maybe we have some combinations with a biological meaning, as the one I'm showing. Okay, then this is just a parenthesis. This is the second part. So we stay mainly with the first one. We'll continue with this. Let me introduce this just to show you a little bit of the complexity. Then, instead of using genotyping data, we're using sequencing data. So the number of pairs increase exponentially. We use more genes. Instead of 67, we use 260. To a point that this analysis, to run this analysis, we need to do this not in a high performance cluster. We needed to use the Mare Nostrum at the BS. Okay. And here the main message, well, we use this to confirm our signal, and this is important because you may say, well, you can have some artifact from genotyping. That's okay. But if you keep having the same result with sequencing, I think you can exclude this artifact. We confirm this signal, as you will see in the next slide. And here what you see is that we are not seeing this very nice correlation. We are seeing this shape. We still don't know how to define this type of plot. If you work with Drosophila, maybe it's a pupa. I don't know. I don't know if I am among Drosophilists today, but it could be a pupa. Okay. Well, doesn't matter. So we expected the straight line. We see this. In this case, what happened is that we didn't exclude comparisons in the same chromosome. Okay. So what we are seeing is very nicely an effect of the linkage disequilibrium. In fact, when you start eliminating the closest positions, this second graph is just to show that this excess of significant positions, so the positions above or below the diagonal, okay, disappears when you exclude positions closer than 6 megabases. Okay, so this is what this graph is showing. Okay, when you exclude positions closer than 6 megabases, the number of significant positions stabilizes, meaning that there is not an effect of being in the same chromosome. Okay, parenthesis. Let's go back to the data because you might be wondering who is participating in this interaction. Okay. And I advance that I will tell you only one of the genes because I'm not allowed to say the second one. Hopefully, I will be able to say this in a few weeks. Okay. Well, this is the case. So this is significant because there is a deviation from the expectation. And remember that we have classified the individuals into 4 categories. Okay. The first one, they are not carrying any of the variants. The second one, they carry the first. No, they don't carry the first. They carry the second. The third, they carry the second, they don't carry the first. And the last one, they carry both, regardless of the genotype. Okay? So we see that the expected categories are not fitting to the observed as expected by the plot that we have there. Again, we validated this with whole genome sequencing data. And then the next step, it's clearly deploying this. Well, I said I can say the first gene. The first gene is the one involved in Mediterranean fever. And the variant that we have detected is this one, one of the founder mutations in Mediterranean fever, in the position 726. And then what we did is displaying this instead of the 4 genotypes, the 9 genotypes. Okay. And what we see is that in the heterozygotes, we see a deviation. Okay. We have an excess of individuals in this category, a lack of individuals in this category. Okay. Remember that Mediterranean fever, it's usually presented as an autosomal recessive, but we have many cases of heterozygote individuals that are affected and many cases where the heterozygote individuals are not affected. Okay, so what we have started to do is checking if this association persists, now not using UK variant individuals, but using individuals in a cohort of heterozygote individuals for this variant. Okay, so somehow this association has been validated in this cohort from the hospital clinic, and now we're in the process of validating in a cohort from here from Bailey. Okay, if it's validated, we will reveal the name of the second gene. Okay, sorry for this. Okay, but let's go to CVID. Okay, so we have applied the same method to CVID, and in CVID, again, this is the first slide I have shown. These are the results that we are having. Let's do some zoom with the hope that we see something and we keep seeing Nothing, okay? So now it's less than 10,000 individuals, less than 1,000 individuals. Maybe if you want, when you move to less than 2,000 individuals, we can start seeing something, although I think this is not very clear. So we don't have a very clear outlier, as happened with autoinflammatory disorders. What we see, if we want to see some pattern, is the second graphs. So the first one belongs to all the individuals, all the genes together. The second one is only for LRBA, because we realize that even if they are not very clear outliers, most of the outliers, okay, in most of the outliers combinations, LRBA was involved, okay? And somehow all these positions here are combinations, for example, of LRBA with TBK1, for example, okay? So again, and this is a very important message, we are not claiming we have here pathogenic combinations. We are claiming that there should be something, okay? So for us, this is smoking gun, meaning that we should inspect this. Another message that I realize now, maybe I have not insisted, our approach has a limitation with the frequency. Even if we have 500,000 individuals, we may have some limitations because of the frequency. If you compare one variant at 1,000 with another one at 1,000, it's 1 in 1 million, okay? So we are still more in the scenario of maybe a pathogenic variant with a modifier variant, for example, as the one that we saw in autoinflammatory disorders, okay? Well, the last graph, I think, yes, here we only report in the last one, specifically the comparisons of LRBA versus TBK1, okay? And to finish, because I know you were going to ask for this, okay, but the number of genes that we have used is 101, okay? 100 and ASIC, if you want, okay? We have done, maybe this is, again, another limitation that we have is the noise. We know, we are sure that many of the signals that we have, they are noise, but we want to find the true signal among this noise, okay? So maybe a strategy should be not going with the global comparison, going gene by gene when you think there is something in this gene, as we have done with LRBA, for example, okay? And of course, you are invited to contact us and let us know which one you prefer to inspect closer, okay? So we did for TASI, and for TASI, well, this is the reality. We have to get nothing. So even at the lower resolution of 400 individuals, we see a perfect correlation, okay? So my apologies for this. And I finish with the very last slide. As I have said, I have wanted to present the digenic model. We are doing something about the oligogenic model also, and this approach is very similar to what you have presented, Andres. Well, also the digenic part, I think it's quite complementary. In this case, what we are doing is comparing a cohort of patients that we have, CVID and diagnosed, where we have WES or genome sequencing and we don't have a clear diagnosis, and we compare this against individuals in the UK Biobank. In this case, it's 10,000 individuals in blue. And what we see in this side is just a count of the number of variants. Then you can adjust by frequency, whatever you want. I think these are the rare variants in two pathways, the cell signal activation and BAF. Because I wanted to report one where TASI is involved, okay? And here the idea is that we don't expect that systematically our patients, the CVID patients, they have more mutations than the controls. No, we expect that the distribution is the same as happens here, okay? But some of them maybe can be explained because of an excess, okay? So what we are expecting is to find some of these dots that it's clearly above the average, okay? Again, we have done this for two pathways. We are developing this for many other pathways involved in CVA, okay? And I hope it was not too long. This is special thanks to all these people, a part of the core lab, this fantastic team at the UB. Thanks also to our favorite collaborations in Vall d'Hebron, and a special mention to Diego Garrido, who is the one who made possible to run the analysis in the BSc Center. And that's all. Thank you very much.
Thank you, Ferran. That was wonderful. And I think that I'm so glad you got invited because this is a bit different for us. Probably some people haven't heard Hardy-Weinberg or linkage disequilibrium since university days. So it's nice to have you here. I'm a huge believer that intersectionality really brings us far away. And this event was all about like advancing and thinking outside the box. So it's super nice to have you here. Also, we love your plot. So, yeah. I think that we're running a bit late. I will give the floor to everybody here in the audience if you have any questions. Okay, no questions. It is so clear, everything. And my question to you was, you are developing this model now for CVID, trying to, I think it's, for now, we do not have positive results, but I think it's like a good idea as we have seen that there are not that many monogenic patients, so we need to keep pushing and exploring. Do you think this is a model that could be applied to many other immunodeficiencies, or do you think that only in the ones that have lower rate of monogenic disorders makes more sense to apply it? Or because I'm asking that because you mentioned that it could be also like this modifier effect. So maybe in the ones that we already know that this is the causing gene, we could have this diagenic effect also underlying.
Can be applied to any group of disorders that you would like. I think that CVID is especially interesting because maybe of the relatively low ratio of success in the diagnosis and because of the heterogeneity that maybe makes think that we have some of these contributions. No? So I think CVID, maybe it's too heterogeneous, okay, but it's a good model. But of course, other PIDs, I think they fit perfectly. I mean, unfortunately, we don't have almost any disorder that has been completely diagnosed. No? Yeah. So I think it's worth, especially also thinking in cases where we have the diagnosis, but to explain the differential expressivity. So these heterozygous patients for Mediterranean fever, we are not looking anymore for the second hit. Well, we are looking for this, but we are not worried. We can explain this because they are heterozygous. But it's nice to find, perhaps finding a mutation that explains why some develop and some others don't develop. So I think we have, there is room for every combination.
Great. That was very nice. We are a bit late for the coffee break, but we still have a coffee break. So maybe we can have 20 minutes and be back at 4:40.
Yeah.
Thanks again, Ferran.
Thank you.
Okay.
Yeah.
Really, the work on common variable immunodeficiency started maybe over 10 years ago when we were approached by a clinical immunologist who was working in Barcelona, Eduardo López Granados, who actually approached us. And you'll see a little bit the beginning of the story, but we've been expanding this topic in the lab, and actually you'll see that it's becoming sort of a central work that we are doing. So I will go briefly over these slides because I think we had enough introduction, but I'll try to sort of highlight or emphasize the aspects that are relevant or interesting to us. So one of the aspects that anyway, here. Okay. So one of the aspects that we are oops. Anyway, I'll do it with my finger. So one of the aspects that we are interested in relation to primary immunodeficiencies and in particular, or primary antibody deficiencies, actually is the wide phenotypic expressivity. So, and I think this is a common problem for clinicians that identify patients, for instance, and the most common manifestation for patients with inborn errors of immunity with primary immunodeficiencies is the recurrent chronic infections, but then there's a sort of array of other complications that actually make very difficult to identify patterns or even families with different sort of subtypes of immunodeficiency and with very heterogeneous immunological features as well. So that's one aspect. And of course, we also heard a lot about today already about the genetics of primary immunodeficiencies. And even a condition like CVID has a very different type of patients. Some of the patients are still without a genetic diagnosis and maybe without a clear monogenic condition at all, whereas other patients have these rare monogenic defects that actually lead to the development of CVID. So, well, this is just examples of primary immunodeficiency, and of course, you know that CVID is clinically and genetically heterogeneous and is mainly characterized by hypogammaglobulinemia, and severe, in many cases, associated with a severe deficiency in switch memory B cells. And I'm not sure about the current numbers, but I believe that is less than 30% of the patients have monogenic gene defects and in a high number of different genes. One aspect that is also relevant to the research in my institute is that there's also some connections or some predisposition in some of the patients to develop hematological malignancy. This is relevant also to sort of the work that we do at the Josep Carreras Leukemia Institute. So also we saw these slides before from this very nice review by Xiao P and Andres. And what I'm trying to highlight here is actually that many different genes, more than 70, are associated with common variable immunodeficiency. So that gives us an idea of how, well, the implication of these genes, which in some cases are T-cell specific genes or more relevant in T cells, whereas others more relevant in B cells. Also that some of the genes that are mutated in CVID in some cases are related to the differentiation in the bone marrow, whereas other genes are more relevant in the germinal center reaction. So actually we see an array of genes, immune cells, and functions related to the development of a condition that actually initially is characterized as a sort of umbrella that covers all of these patients. You can correct me if I'm wrong because I'm here to learn as well. So I'll be happy to, if you interrupt me and I'm saying something that is not exactly or updated. So the idea is again that still a majority of the patients with CVID have no mutation. So that suggests that perhaps either we failed or we were not able to identify mutations in these patients, or maybe there are alternative pathogenic mechanisms. And also, is it interesting to identify perhaps a common molecular umbrella to CVID? Or maybe what are the specific molecular signatures that are associated with different CVID subtypes or associated with different mutations, or whether we can identify pathways that, even though some patients will not have mutations, can link them to some of the monogenic conditions. So one of the initial questions that we had is whether perhaps epigenetic alterations could provide that sort of molecular umbrella. And this is what we started to do over 10 years ago. And in relation to epigenetic alterations and modifications, the main or best studied epigenetic modification is DNA methylation. So DNA methylation is a mechanism that actually is able to switch on or off genes. So perhaps provides an alternative mechanism to mutations, which are characterized by gain or loss of function. So DNA methylation is a potentially attractive mechanism to associate with the loss of or gain of function. And one of the things that, that is interesting with DNA methylation is how is it targeted to a specific genomic region. So DNA methylation does not occur in a given section of the or region of the DNA randomly. It's targeted, so transcription factors are actually major elements in driving where the DNA methylation or demethylation needs to occur. So when we are actually profiling DNA methylation, we can learn about the transcription factors that brought DNA methylation to that specific site or remove it. So that's why DNA methylation is not only relevant per se, but also because it gives you clues of where, of which transcription factors or signaling pathways are implicated. So this is what we can learn also from DNA methylation. So, and for instance, this is a cartoon from a paper published some years ago also by the group of Iñaki Martín Subero here in Barcelona. And we can see that actually DNA methylation changes during B-cell differentiation. So we can see here different stages of differentiation of the B-cell lineage. And we can see that the DNA methylation profiles change from hematopoietic stem cells to late stages of B-cell differentiation, and that this is very specific. So these profiles change in a very specific manner. This importance, for instance, of the DNA methylation profiles is also exemplified by a paper that we published also some years ago in collaboration with the team here in Vallebron and other collaborators like Leonard Hammerstrom or Bodo Grimbacher. So in this paper, actually, we studied DNA methylation profiles of B cells from patients with mutations in activation-induced deaminase, which is mainly relevant in class switch recombination or somatic hypermutation. So these were patients with hyper IgM2 type 2 syndrome. And actually, we found that some of these alterations in DNA methylation in relation with mutations in AID occur also prior to the germinal center reaction. So this is just here to illustrate the role and the importance of DNA methylation in proper B-cell function. So in relation to CVID, our initial study that we initiated in collaboration with Eduardo López Granados and also Carlos Rodríguez Gallego, started with the work with a pair of monozygotic twins discordant for CVID without pathogenic mutations. And initially, we analyzed the methylation profiles of total CD19 positive B cells, perform methylation analysis with an expensive beta array. So actually, it was quite easy and to perform. And then we studied a cohort of CVID patients from Hospital La Paz in Madrid. So in that study, which was published more than 10 years ago, we were able to see that in these total B cells and in the comparison of these two twins, we were able to see a profile of hypermethylation. Also, some genes were hypomethylated in the CVID sibling, and actually those genes that were hypermethylated when looking at total B cells, when splitting and sorting the cells from the cohort, we were able to see that this hypermethylation profile was mainly observed in the memory B cell compartment, and particularly in switch memory B cells. So that was also associated with some changes in transcription. And sometime later, so we decided to exploit this sort of observation, and we expanded the study by using single cell approaches, and also starting with the same pair of twins and a new CVID cohort. We sorted the cells, performed DNA methylation profiling in a single cell with a single cell approach, chromatin accessibility, and transcriptomics, and then also perform amplicon sequencing to analyze DNA methylation alterations in the cohort. So this study, so maybe some of you know, so I'm just going to try to go as briefly as possible. Basically, we confirmed the initial results in this sort of low resolution initial study. And again, we mainly saw these changes in methylation in the switch memory B cells. So here we have the two twins, the healthy one, or at least the non-CVID, where we can see this demethylation profile that is consistent with what is observed in normal differentiation. And here we saw this impairment of demethylation in the switch memory B cells in the CVID twin. So that these are just examples of genes that undergo demethylation in the switch memory B cells and are sort of impaired for demethylation in the CVID sibling. So in that study, we were able to see, when looking at individual cells, that actually this impairment for demethylation is not sort of constitutive. So some of the cells are able to demethylate, and we can see it here. So these are profiles of individual cells. So we can see that, and in light blue, you can see that some of the cells demethylate properly, whereas others are heavily impaired for demethylation. So it's not a property that has been lost in these individuals. It's a property that is not properly, it's a demethylation event that is not properly targeted in all cells. And also what we see, so what we have to keep into account is that, so these patients not only have a lower number of switch memory B cells, but also the few cells that they have, some of them are heavily altered at the DNA methylation level. So the idea was to explore whether these alterations had a reflection at the transcriptomic level, at the expression level. And I'm just going to jump to this, and this is just the validation in the cohort. So I'm just going to jump into this. So here, what we saw, when we perform a transcriptomic analysis, also using a single cell approach, we were able to see that actually there were no changes in expression in the peripheral blood B cells. That was a bit disappointing because those alterations at the DNA methylation level didn't have a reflection at the transcriptomic level. So from a functional point of view, it suggests that, okay, these cells have a aberrant signature of DNA methylation, but they seem to express the genes normally. So, but what we also observe is that the genes that are hypermethylated, actually, if you look at the corresponding transcription levels in different cell types from public data, we can see that actually have only higher expression levels in germinal center B cells, not in switch memory B cells or plasma cells. So that suggests that those alterations that we see at the DNA methylation level in the peripheral blood actually might not be visible in the peripheral blood, but somewhere else. And that's why we decided to stimulate PBMCs from the CVID and the healthy individuals, stimulated and trying to sort of mimic the germinal center reaction, super simplified, with two different activation methods by using CD3, CD28 and stimulated T cells, or CD40 ligand IL-21. And then we performed single cell transcriptomics. And when we did that, we were able to see that the very same cells that did not show alterations in baseline actually had more than 3,000 changes when they were activated, suggesting that what we see in this sort of in vitro experiment reflects alterations that perhaps occur in the germinal center, but we didn't just have the material to identify that in the peripheral. And then we were able to correlate some of these changes with the alterations at the DNA methylation level. Also, what we did is actually take advantage of having generated data, not only for B cells, but also for T cells and other compartments like myeloid cells and NK cells. So this is B cells, but then of course we can inspect the others and identify expression changes in the other compartments. And there, when we did that and using a tool to identify alterations in the communication between cells, we managed to see that actually the alterations in expression in many, am I okay more or less? I'm okay. In alterations in expression in B cells were actually associated with changes in ligands and receptors of T cells and NK cells, and those occur mainly in activated memory B cells. So we see lots of alterations in the communication between ligands and receptors only when memory B cells are activated. We can see very few events in naive B cells or in memory B cells at a steady state. So that helped us to sort of, this is a validation in the cohort, and we were able to sort of reconstruct potential alterations that are taking place and also are linked with what it was mentioned before about the fact that actually CVID is not a B cell disease per se, it's actually involving other cell types, as we, for instance, saw also alterations in the T cell compartment or NK cell compartment. So these are some of the ideas. Also, in relation to these studies, we have also investigated how CVID patients respond to infections. So this is something that we started at the beginning of the COVID pandemic, in collaboration with Klaus Warnatz and Roser Bento. And in that case, we took advantage again of these single-cell technologies to explore how the infection, the initial question was whether SARS-CoV-2 was a particular threat for patients with immunodeficiency, and based, for instance, in the fact that CVID patients seem to experience a slower recovery, or the fact that these patients actually do not achieve a full response to vaccines. So in this case, we took advantage of a very nice cohort that Klaus had developed with patients that they had already collected sample before the infection, and then super systematically had collected samples during the infection and then after the infection at different time points. So actually, there was a really nice prospective cohort with time points before, during, and after. And we were able to also obtain data from healthy donors, so we could actually generate single-cell data and inspect the impact of the infection, how the patients dealt with their immune system to respond, and then their recovery. So I'm not going to show this data. This is also published, but just a very, very cartoon to summarize these results that you can check in the paper is basically that actually these patients that actually we were not able to obtain patients with severe COVID. So and also many reports over that period of time didn't show that these patients, perhaps because they were initially protected, but then they didn't seem to be or to respond worse than healthy people. But actually, when we looked at their the responses in their different immune cell compartments, we saw, for instance, a persistent type 1 interferon response in the different compartments. And then, for instance, for the myeloid compartment, for instance, we observed a persistent activation of inflammasome genes that persisted weeks and months after the disease, or, for instance, other compartments were also affected. So in this regard, sort of the summary of all this is that basically that patients with non-monogenic CVID, which is the one that we have studied so far, not only have lower number of memory B cells, but also profound alteration at the DNA methylation and transcriptomes, and our results suggest that these alterations might occur at the germinal center, and it could be associated with an altered cell-cell crosstalk. And finally, what we also see from these studies is that these alterations in non-monogenic CVID go beyond the B-cell compartment, and actually we see alterations in other compartments, and in the case of responses to infection, we see, for instance, persistent activation of the inflammation-related response. So what we are trying to do now, and it's something that we've started more than 2 years ago, but it's work that is in progress. So I'm just gonna explain a little bit what we are doing now in relation to this. So our idea is now to actually look at monogenic forms of CVID. So we believe that this type of studies that we are doing with monogenic forms will actually provide clues about relevant components in CVID that then can be mirrored into the non-monogenic forms. So we think that if we perform the same type of studies in, for instance, patients with mutations in FKB1, Icaros, or TASI, or PIK3CD, etc., those alterations at the DNA methylation or transcriptome level perhaps can be mirrored into the non-monogenic forms in an expanded cohort and see whether we can better classify the patients from this point of view. And we have, well, this is sort of just to illustrate the relationship of different pathways that are relevant to CVID, like the NF-kappa B pathway or the Icaros-related mutations. We have also included here the JAK-STAT, which is maybe less relevant, the mutations, but this is an example. And these are just a summary, and you know here the mutations that we are collecting. So basically, we have already generated single-cell data for patients with mutations in NF-kappa B1. We have here some of the patients that we have included, and we can see that with this sort of idea, we are actually inspecting the effect of patients with mutations in the different domains of the protein. So this can also be associated with, we are doing the two kinds of experiments of single cell, but also trying to see the binding of this factor to the genome. So see how these alterations are mistargeted, connecting with what I explained before, that these transcription factors are going to be targeting DNA methylation at specific sites. So we have already set up the conditions to inspect the defects in binding of different mutations, and also we have generated the data for the single cell. We have also collected samples, and these come from the mutations in Icaros are even more rare in relation to CVID or immunodeficiency, but we have samples, for instance, from Carlos Rodriguez Gallegos, Xavi Solanic here in Barcelona, or Sergio Rosenbeit in NIH, and also Maria Tapia Torres in the Canary Islands. And also with the idea, we have also some samples from Japan, so we are trying to cover all the different sort of main clinical phenotypes of the mutations in Icaros. So we have mutations in the DNA binding domain, the dimerization domain, gain of function mutations, etc. And finally, we also have generated already data for single cell data for patients with mutations in TASI, and we have here the mutations that are already included in the study. And so the idea is that maybe next year I can present this in the context of what we have generated before. So hopefully we will be able to present that very soon. Another aspect that I wanted to mention today is that actually we are also interested, and maybe this is interest of the team here, we are also studying effects of immunodeficiency in patients with different hematological malignancies. So many patients with leukemia or patients with leukemia that have immunodepleting therapies, so they suffer from secondary immunodeficiency. That's on one hand. And there's also some teams that are investigating the possibility that some of these leukemias, like CLL, actually come from a primary immunodeficiency. And teams like, for instance, Silvia Sanchez in Madrid are studying that possibility. So we have already generated a cohort of patients with secondary immunodeficiency or perhaps a putative primary immunodeficiency and CLL, and we are trying to compare the patients with CLL without immunodeficiency versus those without immunodeficiency to see whether by inspecting the cells that are not leukemic, we can identify traits that actually help us to see if this has any relationship with what we find in CVID or other primary immunodeficiencies. So I think I'm just going to leave it here. So we have also some preliminary data, but it's too preliminary. So also hopefully we can show that in the future. And just to finish, this is the team in Barcelona and our collaborators. I just wanted this is not only to show that we have some money to pay these experiments, but actually all these projects are to work in primary immunodeficiency in the team. So our commitment on investigating these aspects are actually is very high. And for instance, two of these projects, the Emerge project is a Marie Curie network that we are doing in collaboration with teams in Freiburg, in different laboratories in Europe. And all these laboratories are actually devoted to investigate these aspects in relation to primary immunodeficiency or other borders of immunity. And this other project, the PROMISE project, is another coordinated project that we coordinate here from Barcelona. And it's also interested in connecting what we can learn from monogenic immune diseases and see how this perhaps can help us understand complex disease. So hopefully we will have a lot of more things to show very soon. Thank you.
Thank you, Esteban.
Thank you.
So thank you, Esteban. So is there any question on the floor here? Go ahead, Andres. Do you have a mic,
Raul.
please?
Hi, Esteban. So great. Great talk. So I was in the last part when you in these last experiments that you're doing or planning to do, and you selected those patients that you've shown already. You have already some patients with the monogenic CVID. So yeah, if we can call him monogenic, right? Because if so you put NF-kappa U1, which I guess of course makes sense. There are many patients like that you can get. Didn't you think of maybe another gene that can have more right to be called monogenic, like with higher disease penetrance? Because in NF-kappa U1 still maybe 70 to 80% of the patients, so or 20% of the patients.
Which are
NF-kappa U1.
Yeah.
So maybe 20% of individuals carrying mutations do not develop disease, right? It could be similar to TASI. The penetrance is also a bit lower. But so that would be my first question. Do you maybe have another gene in mind that you could use for this with, if you want to make sure you're calling monogenic, and second, if you continue with NF-kappa U1 and TASI, would it be interesting, do you think it would be informative to include mutation carriers that are not affected? So to see also the differences in this signature.
Yeah, so for instance, for the TASI and the NF-kappa project, we have also even for the Icarus project, and we are doing this in collaboration with the team here. So we are basically including the patients that are available and the patients or families that are actually interesting. So we have some asymptomatic individuals in the cohort. So we are going to be able to interrogate the effect of one mutation versus another in different domains, but also why, or try to understand why some patients or individuals with the same mutation develop or not. Another consideration is maybe because in most cases it's the mutations in one allele, so it could just be an effect of which is the allele that is mainly expressed, no? So we need to test that also to assess that. But but at least with the profiles, we will be able to see how the different immune cell compartments are correlated with the clinical manifestations or the absence of manifestations.
So for the NF-κB1, so there's this disease activity score, right? That I think it's being published soon. Or so, would you take that into account?
Sorry.
So there's at least a work. I don't know I mean, it's been led also by Bodo. So this disease activity score. I don't know if you mentioned that. If you would take that into account, I mean, in addition to affected, not affected, but maybe the severity of the disease.
Yeah, we will do that. Also, I forgot the question about the different other genes that we are considering. I mean, in the end, it's a matter of resources, no? Other genes that we are considering, and we have been discussing just 1 week ago with Bodo and the other teams that are part of the PROMISE consortium. So CTLA-4 is one of the ones that we are considering. We are considering, although maybe it's a little bit on the side, no, because it's not, but ADA2 are mutations that we are considering. It's difficult because the number of patients is lower, but PIK3CD would be really cool to look at, and also as a reference for many. So, but yeah.
Thank you. Okay, so we need to move on, but we can continue the discussion with the roundtable after this. So our last speaker is Janire Pereurena. It's my pleasure to introduce her. She has a PhD in immunology. She is the reference person for all the cytometry in the routine lab, and she have the pleasure to co-run the research lab with her. So Janire, go ahead.
So thank you for the presentation and for the invitation and for all the attendants that are still here on a Friday afternoon. So what I am going to present in this talk is a challenging familial case of a suspected antibody deficiency. That it's the practical example of what complexities we found in the clinical and molecular diagnosis at a hospital in patients that are suspected to have CVID. So first, I would like to begin presenting the clinical case of a child that was born with no complications. But when he was 6 months old, he started to present recurrent urinary tract infections despite having a normal renal anatomical evaluation. And when he was 1 year old, he developed abdominal septic shock, and that conducted to a more in-depth analysis that led to the detection of severe neutropenia and hypogammaglobulinemia. So the neutropenia was treated with a brief course of GSCF. And he was started on immunoglobulin replacement therapy. We will see why after, because he was quite small, but it will make sense after. But when he was still 4 years old, he remained with this quite severe neutropenia, even when he was under treatment with this GCSF. So that led to a genetic testing. And in this genetic testing, what they found, sorry, it's a little bit uncomfortable.
You can,
I don't know where to look.
No, you can go on the right. It's going to be easier.
Yes. You can hear me now? Yes?
Yeah.
Okay, sorry. So in this genetic testing, we found a CD40 ligand variant. And this variant was classified as pathogenic because first, it was a predicted null variant in a gene where loss of function is a known mechanism of disease. And you can see here on the right the effect that this variant has at RNA level. So you can see the reference wild-type allele, and the mutated allele that this variant will cause is two different transcripts. An exon 2 skipping of exon 2, it's one of these transcripts, and the other one will be the retention of intron 2 with a new stop codon on exon 3. So this means that you are not going to have a normal protein in these patients that are hemizygous to this variant. Moreover, this variant was extremely low. It had a extremely low frequency in the database of general population. Moreover, it was already described with well-defined functional studies showing that it was deleterious for the protein that it's codified by this gene. So therefore, an hyper-IgM syndrome was diagnosed in this patient, and he was started on antibiotic prophylaxis, awaiting for a hematopoietic stem cell transplantation. And in this moment is when the patient arrived to us, because we are a national reference center for hematopoietic stem cell transplantation in inborn errors of immunity. So everything that I have said before, it was done in other hospitals. And after the transplant, he did some complications, but after 4 years, he's off treatment and he's doing quite well now. So when we think about hyper-IgM syndromes, these syndromes are characterized by a marked decrease of IgG and normal or elevated IgM. And this is because there is a defect in class switch recombination. And all of you will remember from the basic immunological class that when a naive B cell gets activated by a T cell that presents the antigen, they will form the germinal center reaction, and there in the germinal center reaction, you will have the somatic hypermutation and also the class switch recombination. And this class switch recombination is mediated by an enzyme that is the activation-induced cytidine deaminase that will process this recombination of the germline transcript, that it's the immunoglobulin transcript. And if you don't do that, if you cannot do the class switch recombination, you will only have IgM and IgD transcripts. That's the naive B cells. But if you do this recombination, you are going to be able to produce also other isotypes. So in these cases, the patients with hyper-IgM syndrome are not able to do the class switch recombination and will only have IgM or IgD that it's not secrete so much. So there are a lot of defects that can give rise to this syndrome because there are a lot of pathways that induce this class switch recombination and somatic hypermutation. Here you have a graphical summary of all the defects that have been involved in these syndromes. There is a star in all the genes that have been described to be associated with these syndromes, but the one that it's the most prevalent is the one caused by CD40 ligand deficiency, that it's located, this gene is located in the X chromosome, and that's the reason that we talk about X-linked hyper-IgM syndrome, and accounts for the majority of cases. So CD40 ligand, it's a TNF superfamily protein, and it's normally expressed in activated CD4 cells, mainly in the follicular germinal center follicular T cells, and it interacts with CD40 in B cells, but also in other cells, and signals through TRAF proteins to give rise to different pathways, but the one that we are more interested in right now, it's in the class switching. So without this protein, we cannot have this class switching. About the clinical manifestation of X-linked hyper-IgM syndrome, you can see that almost all patients present with pneumonia, a lot of them with infection of pneumocystis jirovecii. A lot of them present also with upper respiratory tract infections and neutropenia and oral ulcers. And also it's quite important that there is a significant number of patients that present with hepatitis and sclerosis cholangitis, that it causes high morbidity and mortality in elder patients, and a lot of times it's associated with Cryptosporidium infections. So all of you know that predominantly antibody deficiencies don't associate with these kinds of pathogens because are more associated with encapsulated bacteria infections. And this is true because in reality, X-linked hyper-IgM syndrome or CD40 ligand deficiency, it's not a predominantly antibody deficiency, but a combined immunodeficiency. And this is because CD40 and CD40 ligand interaction is not only important for class switch recombination, but also for the licensing of dendritic cells and activation of cytotoxic T cells, and also the activation of macrophages and neutrophils. And that's the reason that we are going to have these kind of infections that are not typical of, are not encapsulated bacteria, like in a CVID, for example. So after this brief review of hyper-IgM syndrome and CD40 ligand deficiency, we are going to go back to our clinical case. So you remember that we have this child in which we found a pathogenic variant in CD40 ligand deficiency, and in that moment, it was performed, the test was performed also in the familials, because there was interest on where this variant come from. In the familial segregation analysis, the variant was also found in the mother. And interestingly, the mother was a 40-year-old woman that was diagnosed with CVID in her teenage years due to recurrent pneumonias and hypogammaglobulinemia, but she also presented with abdominal tuberculosis when she was 4 years old. That is not so typical of CVID. But they started with immunoglobulin replacement therapy when she was a teenage, and she has remained asymptomatic since starting this therapy. However, as we saw this variant, we thought that we could be in front of a possible monogenic cause of CVID in the mother, so we decided to do molecular testing, more in-depth molecular testing on the mother. So for this, we need to review other mechanism that it's important, and it's the chromosome, the X chromosome inactivation. So all of you probably know that females physiologically in our early divisions in the cells that early divide after the zygote, we silence one of our X chromosomes to ensure that our dosage of X chromosomes is compensated. So, and this silencing that is mediated by epigenetic modifications is transpassed to the descendant cells. So in a normal female, in a healthy female, you will have that more or less half of our cells express one of the X chromosomes and the other half will express the other X chromosome. But in a lot, it's not rare to have healthy females that have this pattern skewed in which there is an overrepresentation of one of the X chromosomes, and normally this is not, it doesn't have any clinical consequences, but if you have a pathogenic variant in an X chromosome, and this is the one that is overrepresented, you can have a disease like a disease that is normally associated with males instead of females. This has been reported in inverse for immunity. A lot of years ago, it was already presented for Bruton tyrosine kinase deficiency. And it has also specifically been reported in CVID. So we thought that that could be happening in our patient. So the test to check if there is an skewed X chromosome inactivation, the gold standard assay is human androgen receptor HIMAR assay. We did this in our patient, and we confirmed that there was an skewed X chromosome inactivation in the mother with a ratio of 14:86. But with this assay, we cannot say if the chromosome, the X chromosome that is overrepresented is the one that is wild type or the one with the pathogenical variant. So we performed a molecular assay sequencing of the RNA to check if the pathogenical variant was the one that was overrepresented and was the one that could explain her clinical phenotype. So remember that the pathogenical variant will cause two different transcripts, one with an exon 2 skipping and the other one with intron 2 retention. But in the case of the mother, we are going to have these two transcripts and also the wild type. Okay, so when we did the sequencing in the junction of the exon 2 and exon 3, we could see three different transcripts that you can see in the figure at right. And you can see that some are signaled with a circle, and these ones are the ones that are different in the three transcripts and are the ones that are corresponding to the wild type allele. So you can see that the height of these peaks is quite lower than expected. It should be more or less half of all the cDNA material, and you can see that it's quite low, almost representing 5 to 10%, although it's not quantitative. So we confirmed that, in fact, there was an skewed X chromosome inactivation in the mother, and that the X chromosome that was overrepresented was the one with the pathogenical variant. We also wanted to check if this was presented at protein level. So we did a functional testing on the mother. We isolate PBMCs, and we incubate them during 4 hours with PMA and ionomycin to activate the T cells so they can express CD40 ligand, because CD40 ligand is not activated in resting. And we did flow cytometry staining and analysis. we did the assay in parallel in a healthy donor to be able to validate the assay. So here you have the flow cytometry results. You can see that the healthy donor, this is gated on CD4 T cells. You can see that the healthy donor expressed CD69, that it's an activation marker, so all the cells have been activated, and almost all of them present a CD40 ligand, that it's CD154. But in the case of the mother, you can see that the activation has been correct. Almost all cells express CD69, but there is a significantly the reduction in the expression of CD40 ligand. So we confirm that the mother is not expressing CD40 ligand as a healthy donor. We also perform the extended immunophenotype B and T cell immunophenotype. And what could we see is the normal pattern that we will expect in a CVID with very low switch memory B cells, normal CD4 T cells, and a normal representation of naive CD4 T cells. So we could say that this patient fits into the criteria of CVID diagnosis by ESID, because by this criteria, the definition of a profound T cell deficiency that should be excluded is to have normal T cell numbers, CD4 T cell numbers, and a normal representation of naive CD4 cells. But we know that for CD40 ligand deficiency, you can have these numbers in the normal range, but you still have a defect on T cell, and it's a combined immunodeficiency. So all of this testing led us to a new dilemma. This patient has a CVID, a monogenic cause of CVID, or does she have a CD40 ligand deficiency that it's a combined immunodeficiency? And this is a real dilemma because it could mean a change in the management of the patient, because with immunoglobulin replacement therapy, it will be not enough for a patient with CD40 ligand deficiency, because as we have said, they present with infections by pneumocystis jirovecii and also a lot of mortality and morbidity due to cryptosporidium infections, and also they have these severe neutropenias. So right now we are still discussing because it's not so easy. The science is not easy to translate to reality because the patient has been asymptomatic for more than 20 years only with immunoglobulin replacement therapy. So for now, it's only close monitoring. As conclusions, female patients with suspected variants in X-linked genes known to be associated with CVID or CVID-like disorders should be tested for their X chromosome inactivation status. Identification of a skewed X chromosome inactivation pattern with predominant expression of the mutated allele can provide insight into the causative. I think they are already ending. Into the causative or contributing role of these variants in the CVID-like phenotype. It's not me. Depending on the degree of skewed X inactivation and the resulting expression function of the affected protein, a change in patient management should be considered based on the specific gene involved. Last but not least, thank you to all of our both groups, the Translational Immunology Research Group and the Infection and Immunity in Pediatric Patients Research Group, because a lot of people has worked on these two patients to be able to resolve the cases and also to give these patients the best patient care options. And thank you.
Thank you very much, Janine, for a great talk. So I think that, in fact, you may have already answered a question that was on another talk previously, which is at what time of the patient journey should you do a genetic test? No, and I think that's this case really exemplify the fact that from the beginning you need to test your patient, no, because it might really change the management of the patient and maybe of his mother or another sibling, no? So thank you for that. I don't know if there is any question on the floor or online. You remember that you can put your questions online. If not, Laura?
If not, we'll thank Janine again. And then we move to the roundtable. So we invite all the speakers to join us. And also, if we can go back to Xiao in the screen, that would be nice. We'll move to one side, so it's a bit more. You can come up. Maybe we can do a bit of a circle. Yeah. No, jo vaig aquí, que així... Sorry, on vulguis, és el joc de les cadires. Sí. You can go here. No, Ferran, you can go here. Yeah. Okay, so the title of this roundtable is Shaping the Future, Challenges and Opportunities in CVID. And we prepared some seed questions, but first of all, I'm going to give the opportunity to people online. This is your last chance. I'm going to look at the camera to submit your questions. And also people here, if you have questions, now is the moment. Also for people here, if at any point you want to say something, comment on something, just please raise your hand and we'll hand you the mic. And I don't know if we can connect back to Xiao. Okay, no worries. Xiao, if at any point you want to join, you're very welcome to do that. So, yeah, first of all, I don't know if any of the speakers has some general comment. We've been hearing a lot on different aspects of CVID, from clinical to evolutionary, genetics, moleculars. Any general reflection?
Yeah, maybe. So I was thinking during all the talks that I have the impression that B cells, which are the target cells in CVID and in other primary antibody deficiencies, are like fragile cells. So because, you know, most of the inborn errors of immunity are in the group of primary antibody deficiencies. And we have seen that not only monogenic mutations, but unknown genetic defects or genetic modifiers or epigenetics can affect the B cells. So it's because B cells are fragile really, or because B cells really have a hard task because they have to develop in bone marrow. In bone marrow have some checkpoints and selection processes, but when they reach the periphery, which is hard to reach because most of them die on the bone marrow, then they have to recognize antigen, and after that they need to do all this class-width recombination, somatic hypermutation, followed by another round of very strict selection process where most of them die. So it seems exhausting. So this comment is just a reflection because, you know, B cells have a very important function, but to do this function, they must go step by step in a hard way. So I don't know if it makes these cells also more easily targeted by environmental, epigenetic, and genetic defects. It's just a philosophical comment, but I think that these cells are pretty the target of this disease.
Okay, thank you, Roger. So I wanted to ask each of you that present something today a bit. We've seen that really genetic diagnosis or other molecular tools really help to our pieces of a puzzle, no, to really get the disease of patients. So I would like you maybe to just say, where do you think that your piece of the puzzle go? So, for example, I don't know, at what point for Janine you would consider an X-link disease in a woman when you filter maybe some variants or things like that. So I don't know if you want to start, Esteban, and then we can go like that.
Okay, so
for example, for you, it would be at what time, at what point of the disease journey or the molecular diagnosis you think that you have to think, okay, it might be more a thing of epigenetics than maybe a pure monogenic variant?
So I don't know if I'm going to more than respond that. Instead of responding, I'm gonna sort of divagating, no divagating, because
Hi, Xiao.
So I think, I mean, when you're talking about a pathogenic mutation that it occurs in a functional domain, and for instance, when the mutation occurs in homozygosis, which still in some cases doesn't always lead to a clinical. I believe in some cases even a mutation in the 2 alleles doesn't necessarily lead, for instance, in some TASI patients to the CVID, I believe. And at the same time, so I think it's very complex and many mechanisms can account. So I think what is very clear maybe is in some cases mutations almost in most cases is going to lead to the condition, but definitely the fact that there's this phenotypic expressivity where you can see that the same mutation leads to different clinical outcomes. It's giving you the clues that there's many modulating factors that are influencing the outcome, no? And it could be that maybe the gene may have some functions in other cell types. In some cases, it's not for all the cases, not for all the genes, but also that maybe epigenetic mechanisms maybe contribute also in cases where maybe the genetic mutation is very clear, so this can modulate the phenotype. I don't know if that's explained.
Yeah, before passing the mic, I have because I have a question for Esteban from a naive person in epigenetics, or quite naive, which is, I'm always wonder what do you think the epigenetic alterations that you see in your studies, some of them are pretty clear, are the cause or the consequence? Yeah. You know, because when you have a mutation, you assume that the mutation is the cause, and then the cell type alterations, the clinical phenotype is the consequence. But you know, in methylation, you never know if there is a primary methylation defect or the methylation defect that you are seeing is because there are other genetic and recognized alterations or environmental factors that cause this epigenetic. So what do you think?
Yeah, well, I think is that I mean, in the same way that the classical egg and chicken or whatever, no? So okay, this is not possible to solve. I would do the same here. So trying to assign, so I don't think you need to contrapose epigenetic alterations against or versus mutation. I think, so for instance, if you have a mutation in a transcription factor, like for instance, NFK, something in the NF-kappa B. So this is a transcription factor that might, sometimes this protein recruits an epigenetic enzyme and then this causes something. So if there's a mutation or an alteration in the NF-kappa B pathway, this is going to alter downstream epigenetic alterations. So that seems like, okay, this is the consequence of something else. But at the same time, this is in the middle of something that, depending on how high you are in the hierarchy, can have also downstream consequences. So, of course, you want to know what's above everything. Sometimes it's going to be very difficult or even impossible to know. But as the higher it is, maybe the higher are the chances that if you touch this, for instance, by doing some gene editing, which are now available, maybe you can have an impact in what is downstream, and that can have an impact on the function of the cell or the clinical phenotype. So, I don't know if I have responded. But
no, it's just.
So I think my answer will be more practical about what I see normally at a clinical laboratory. So I work normally in flow cytometry, so I interpreted data on flow cytometry, and my colleagues here are immunogeneticians. So if my immunogenetician say me, okay, this patient has a suspicion variant, I will know. I should do the molecular testing. I am sure of that. Relating with the flow cytometry, I think that we can have a lot of important data, because if I saw that this patient has a defect in T cells, like normally patients that are have the name of CVID, but that had a monogenic cause, a lot of time they had defects in T cells, like very low naive CD4 cells, and we have seen that for NFKB1, CTL4, Icaros, so a lot of other patients. So definitely if there is a defect in CD4 cells, I will do molecular testing, like checking it up. And also I think it's quite interesting, and I also think as Roger that B cells maybe are not very robust or not so much as T cells. I suppose that that's evolutionary. I mean, without T cells you cannot survive. With B cells, well, maybe you can survive until having a child. I don't know if very long, but what I see in B cells is that I am really interested in patients that are classified as CVID that don't have almost any B cell. And they don't talk much about that in classifications, because they always talk about the switch memory B cells, but a lot of patients with CVID don't have any B cell, and we are not worried about that. And that's interesting as a, I don't know, scientific society, that patients without B cells can survive until late years. So I will probably try to do more molecular testing on that patients, but I don't know which molecular testing, because I am not an expert in that. So that's my opinion.
Thank you, Janina. Well, in our case, I think we are quite far away from the diagnosis. But before, Roger, you have said something that you made me think that, I don't know, you were insinuating that because of the complexity of the development of the B cells, they are more fragile. So maybe because there are more genes involved, you could explain the heterogeneity of the disorders that we see, something like this, no? So could be explorable, no? So, well, you know that we like correlations, and and it's something that maybe it could be worth exploring. Then on what you have said, well, something that in this case it was Andres in your talk. As I have said, we are not in the diagnosis, so we are exploring novel mechanisms. And something I would like to insist is that we need to analyze also the healthy people, okay? So as we did in the UK Biobank, well, they are not healthy. There's a bias sample, but they are essentially healthy or not enriched for rare disorders, at least. But to assess the effect of a variant, we are used, as you were doing, no? So these carriers of the TASI variant, what else do they have? But maybe the interesting thing is, okay, so the healthy people with the TASI variant, what are they having that maybe it's having a protective effect, okay? And with this.
Thank you. Yeah, so I mean, going back to the piece of the puzzle, I don't know, I want to go back to a couple of slides that Xiao showed at the beginning of the session. So I think we are not lucky enough that, I mean, in immunology, so there's these three components, right? That's how we're showing out: genetics, environment, and stochastic. So basically, yeah, we're not lucky that in immunology, I think there's probably a higher prevalence of the effect of the environment. It's not as simple for us. And I'm saying this, I mean, coming more from the genetics background. It's not so easy for us as it is for other realms of disease areas. And so for me, I think it becomes important as a key piece of the puzzle, the knowledge. So the specific knowledge that we are generating as a community, I mean, in the research setting. So we are, you know, publishing and, you know, generating all these papers, all this information, like, you know, this interesting case that was presented now by Janine. So I think we really need to make an effort to catalog of all of this information, all these genetic paradigms, pitfalls, all these nuances, all these allelic mechanisms that Xiao was speaking about. I mean, what happened with Icaros and all the different mechanisms of action that are for every gene, because I mean, it really has an important impact on management. And yeah, I mean, this is why, I mean, I don't want to advertise or publicize our project here, but
go ahead.
But yeah, this is why we developed this public resource GENIA, the GENIA database, and I think this is where our little contribution comes, I think, to the society, that we're making this big effort to address this gap, this knowledge limitation, and hopefully, I don't know, in a few years, we are at least, because as Xiao said, so that technological gaps or limitations are more easily solvable nowadays, and it's more the structuring of the knowledge and the generation of more knowledge, as we were saying with the TASI variants. I mean, now I guess no one wants to work on TASI anymore, but it would also be useful. I mean, not only for TASI, but in general for any other genes to generate these arrays of functional data, right? I mean, of course, the patient cell assays are also essential, but we have now the advantage of being able to perform these MAVE studies on almost any gene. So I think it would be important to generate this data and to put it somewhere that is accessible to everyone, and this translates into better diagnostic procedures. And of course, I mean, genetics, as Xiao mentioned, is not sufficient, and sometimes you can just basically diagnose your patient with a regular molecular cellular assay, and yeah, we have to think about that.
Thank you, Andres. So maybe for Xiao, we can, instead of asking the same question that we ask the rest of the panelists, I think there was a question that wasn't answered from the chat before, and that is quite relevant from your talk and also from your colleagues' talk, is that considering that the majority of immunodeficiency have a part of multifactorial, let's say, and beside only the variant, should that, as a molecular scientist, be included in the report regarding VUS?
I'm so sorry, Jacques. I'm at the airport, so can you repeat
no problem. Thank you for connecting.
Your question again?
Yeah. So the question was, since there is a piece of puzzle different with IAI compared to other diseases, and the complexity of the immunodeficiency, if it should be reported like that when reported VUSs. So saying that this is a VUS likely pathogenic, but you have to take into account a lot of other things like epigenetics, I don't know, X-skewed chromosome, etc.
Yeah, so I think people really need to be careful about what a genetic test report actually means. So I think this is something I try to really make clear to my colleagues and my patients, which is when you're doing variant interpretation, first of all, it's a very subjective human-made framework that's constantly evolving as we learn more, right? And usually, when whether it's me counseling a patient or Andres writing a report, we usually break it down into gene function, gene dysfunction. You guys have all seen these, right? And what the specific variant actual interpretation is, and then the fourth part is, what does this variant mean in the context of your patient's clinical presentation? So that fourth part is not something technically any kind of lab writing a report should take responsibility for. It's the provider who's counseling the patient and taking care of the patient's responsibility. It's your job always as the provider to take any results and put them in the context of your patient, because really what a test report is it's telling you how much genetic evidence is there to support linking this spelling change to that gene, and linking that change in the gene to a purported gene-disease relationship, right? That's the only responsibility of a genetic testing report. But then you have to be the person to say whether or not it has anything to do with your patient. I get pathogenic variants all the time in genes where I'm like, yeah, that's a pathogenic variant, but I do not give a crap about this one because, it's a common mutation in a gene that confers, let's say, increased risk for atopy and dry skin. That's not why I sent genetic testing on my patient. I want them to know that they have a predisposition to atopy and dry skin. That's great. It probably explains why they have eczema and asthma, right? But that's not the problem I was asked to solve. That's not the condition I was asked to try to make better for the patient. And this is why, again, it's really important not to realize that genetic testing is just another assay, right? So you're integrating all of the data. And one thing no one has mentioned is treatment. So most of our patients, it's nice to have, lots of lovely granular immunophenotypic data, right? But that's still descriptive, and that changes as patients are exposed to different things. You know, when you're testing a patient who is actively infected, you're not going to interpret their type 1 interferon signature the same way as when they're not having, let's say, an EBV or adenovirus infection, right? You're not going to interpret your patient's B-cell profile the same way. So a lot of our patients come to us having already gone through so many cycles of different kinds of immunomodulation and steroids and, you know, everything. It becomes very, very hard to try to go back and guess what their treatment naive etiology was, even though that might be the most proximal descriptive clue to the pathobiology. Oftentimes we don't have that. We only have the luxury of knowing their, you know, their clinical course. So we find ourselves a lot of times really trying to guess what we think the original pathobiology is. And that's, you know, an unfortunate fact of reality. But there's also a lot of things to be learned by whether or not, you know, a person's pericarditis is responsive to IL-1 blockade. There's a lot to be learned when you see that a person's B-cell profile is improving when you treat their type 1 interferon signature with a JAK inhibitor. Those are also things that we have to try to characterize and learn from our patients, as much as the healthy controls and, you know, all of these lovely monogenic disease situations.
Thank you, Xiao. Thank you very much. I think you raise a very important point that we have really on a daily basis in the clinics and in the labs, no, on the functional testing of variants, and that you don't have always the ideal condition of the patient, or once you have the mutation, then you already treated the patient, so you don't have, or you might have a bit of blood stored from before, but it's not enough to do all the experiment that you want. So I think it's really important to take that into account.
Yeah. I also wanted to comment a bit, like nowadays, artificial intelligence all over the place, and linked to what Andres was saying about this MOVDB and all BD and all this initiative to try to test as many variants. We know that there are big labs that can do this arrays and test all the variants in the gene. Some others cannot. But do you think, or how far are we from these in silico predictions being robust enough to be believed? What do you think about that?
Be careful what you answer.
I'm sorry, Andres, you're just next to me.
Yeah, so I mean, the question is how AI could improve in silico predictions.
Yeah,
prediction tools. Yeah. I mean, I don't have deep expertise on how these algorithms are developed. I mean, what parameters they take into account. I mean, the feeling is, I mean, me personally, I'm not taking any of the in silico predictions really into, I mean, strong consideration. I mean, they're just, something else that adds to the, but and there are way too many. It's just like, so that's the first. I mean, which one do you trust more? Of course, you can do a aggregation of all these in silico predictions, but yeah, the thing is that I don't know if I have the feeling that they should be tailored to every single gene, because every gene is different. So, I mean, it's nice. You try to come up with algorithms with a way, a tool that it fits all, but I think it doesn't work because every gene behaves differently. You have different analytic mechanisms, and I don't know. For me, it's something that it's sort of an impossible task. And I don't know if even with AI, I mean, I don't know if this would be possible. Again, I think I'm going to go back to the lack of knowledge. I think if you could feed an AI agent, I don't know, with the right knowledge, created knowledge for each specific gene, maybe you could come up with gene-specific algorithms or prediction tools. But yeah, again, I mean, I'm not so expert in this area, but
No, yeah, I agree, because we have been experimenting a bit with that, and what we realize is just that you need your training set to be focused on your kind of disease, the mechanisms in your disorders, and usually this is not even possible, especially with rare disorders. So it's a bit, sometimes I feel like nowadays it's like AI is going to solve all our problems. But in some cases, we're maybe a bit still far away. I don't know, Jack, if you want to comment on that.
No, I think
we can still keep our jobs, right?
yeah. No, I think there is a big problem with. We have a lot of faith in AI that will solve all our problems, but there are problems that still remain. I mean, for today, for example. You cannot publish a single case of test a CVID with a test imitation. You won't be accepted in any journal, a single case. But this would be useful if everybody has their cases published with granularity, because the one thing that resembles to that maybe is the acid registry, but again, it's not very granular. It's a lot of data. It's the biggest registry on AI, but still it's the structured data with a good quality, but it's not the reality. So and if you don't, I mean, Xiao said before, garbage in, garbage out. It's a bit the same. If you don't have all the tools to interpret, and I think you've done, Andres and other here as speakers, you've done a lot of work in creation data, and it's really a manual job, like really time-consuming and effort-consuming. So it's not that easy.
I think that in the next weeks or months we will see, how you say, an avalanche and an increase, a high increase of candidates to be validated, because with these tools they have liberated, especially this week. I have still not tested, but what we will have is that I think we are increasing a lot the power of prediction for intronic variants or variants in regulatory regions. So yes, we have to rely on functional validation, but I'm sure that now we are going to analyze many patients and we will find candidates in new patients, especially in regulatory regions, that it's where these tools seem to be powerful. So in this sense, I mean, I am optimistic, even if at the end we need the specific knowledge about the disease and the cell type, but and we will need to rely still on functional validation, but maybe we'll get to a point that it's when, as when you find a new loss of function variant, maybe we can find some new variants in regulatory regions that we will assume they have a functional effect.
So meanwhile, Bodo was saying that publish Altachi, this is what GenAI is good for. No, I think so. If you want to answer, Andres, but no, I think that really having more data is easier to interpret and to feed AI models. I think it was the point of the discussion before. I don't know, Bodo, if we made that clear enough, or it was maybe not very well explained by me. Sorry.
Yeah, just a comment on prediction. The problem I see on prediction and with the AI tools is that, at least currently, prediction or AI relies on the information that humans provide. So the problem for some type of variants, let's say the missense variants, is that sometimes we don't even know what is the exactly molecular function of the gene. So we say, okay, this is a transcription factor that acts on this gene. Yeah, but maybe for a good prediction, we should know which residues specifically interact with the chromatin and the amino acid and the DNA nucleotide. So we are far from here for most of the genes. So if we do not provide this molecular information to the machine, let's say, it is difficult that the machine really can predict this. So, for example, for splice variants, the prediction is pretty good in the last years because we provided some real information about the cryptic splice site positions or the splice process. But for missense variants, for structural protein variants,
no
there is
question.
still a work to do, and we rely most on functional assays. And even functional assays have some limitations because usually you choose one readout. For example, we want to test the NF-kappa B pathway. Okay, what do you test? If the NF-kappa B dimer go to the nucleus, or if the dimer is formed, or if the inhibitor can bind the dimer. So it's even difficult. So, and I have a question related with that to Xiao, because you put a wonderful example with your Icaros patient. So, and you showed that Icaros, so the gene IKZF1, have at almost 6 or 7 different genetic mechanisms that cause disease. Haploinsufficiency, dominant negative, gain of function, dimerization. So do you think that this could be possible in other CVID or inborn nerve immunity genes? Because maybe in other genes, we are just saying, okay, there is a mutation in that gene, and we did not take into account that depending on the motif or the region, this mutation or this variant can have different effects. So do you know that this can be in the future more, let's say, probable for other genes?
Yeah, I mean, I think that's already starting to be shown, not just for IEI genes, but also outside of that domain. And again, I think it goes back to what is the actual assay testing, right? So I think the inflammasome genes are another really good example of this situation, because before they developed the high throughput spec formation assays, you know, a lot of the previously tested NLRX variants were thought to be benign, right? Because they only looked at it under one particular condition, and they didn't realize, for example, that there were different kinds of priming and stimulation conditions, just as they didn't realize maybe there are different outcomes where sometimes you don't have the caspase-mediated IL-1 beta and IL-18 production, and you should be looking for, you know, forms of cell death as a readout, right? So that's I think a very good recent example of how we've revised our understanding that we need to assess multiple functions when we assess inflammasomopathy genes. IRF4, I think that's another really good classic example, and I think for almost every transcription factor we're going to have those situations where you're going to have multimorphic function, right? Because transcription factors, the nature of their activity is that they have to be able to bind DNA, they have to be able to transactivate specific loci, and so if they're transactivating the wrong loci, that's automatically, you know, neomorphic of some kind, right? If they're, you know, doing other things like binding partners they're not supposed to be, they're probably doing some weird dominant negative stuff. If they're binding the partners they are supposed to be binding too tightly. I mean, so there's so many different ways because it depends on what the actual function of the gene is. So I totally agree with you, Roger, when you were saying, you know, we really need, when we think about, you know, how to do mechanistic discovery or how to predict mechanistic effects, we need to give more instruction, I guess, to our discriminative models about actually thinking about what the gene's function is. And certainly, you know, one really old example is Noonan syndrome. So, you know, forget the complicated immune genes. Like if you take something like just PTPN11, there's plenty of experts in the RASopathy field who cannot, for the life of them, agree on whether a specific missense change is gain or loss of function depending on the assay readout. And I had to be in this situation because we were having a big argument about whether or not to put a patient on a MEK inhibitor versus a different kind of kinase inhibitor. And it depended on whether or not we thought it was gain of function or loss of function. And at the end of the day, everyone just threw up their hands and said, just give the kid everything because the kid's dying, you know? So yeah, I think this is going to become more the rule than the exception, because we're going to be able to develop more and more different kinds of assays with different, more specific readouts for function.
That's very nice, good and positive note to close the discussion. So, yeah, we have very limited time to finish. So I want to thank everyone. I made the list. First of all, to all the speakers for being here, for your expertise, your insights. I think it was a very stimulating discussion. Also to the audience. especially the ones that are here in person on a Friday, almost evening. So thank you, and people online also for joining us. Special thank you to our sponsors, the Barcelona PID Foundation, who has been helping a lot on organizing this. And I really hope you leave today, have learned something new, inspired by the conversation. And we really hope to organize this session again in another topic next year. So we'll be very happy to welcome you. And finally, Roger, as a head of the Translational Immunology Research Group, is going to close the event. Thank you.
Okay, thank you, Laura. So it's been a great session. Honestly, I enjoyed all the lectures. I think all the audience enjoyed the lectures. So I have nothing more to say. Just maybe what I thought during the lecture is like a metaphor about CVID. Let me explain. So I think that people is working in CVID are like gold seekers. So people like are looking for gold. They use these filters, you know, sieve is the word. I look it in the translator because I didn't know. So when they use these sieves, these filters, I imagine that the patients are at the top of the filter. These are patients clinically diagnosed as CVID. And then we put a lot of efforts to move the filter using genetic technologies to find the mutation. And each time we find the mutation, the one patient drops the filter because it's no longer a CVID, it's an NF-κB1, LRBA, etc. So we put a lot of effort to take out the patients of this sieve, but the remaining patients are very helpful because are those that are genetically negative. And instead of failure, these are the patients in which we truly have to prove these models like the Ferran model or the epigenetics model. Yeah, because sometimes you take patients that are genetically negative, but maybe are simply genetically undiscovered. So this really discovering the genetically positive patients make the non-genetically positive more pure, and probably in this group, the non-monogenic models are worth to test, and probably the signals will be higher. So anyway, thank you very much, everyone. It's been a great session, and for sure, CVID will continue to be the topic of a lot of sessions. Thank you.
Thank you.
Thank you. Molt bé.
Bye-bye, Xiao.
Bye.